Yes, yes, thank you. All right, well, good morning, everybody. Some people must still be on their way down. But welcome back to day 2 of this great meeting. Mike and I are going to try to do better on the microphone this time.
Yep.
You're number 1.
Okay. So good morning, everybody. I hope you had a nice evening, and looking forward to today's really packed and I think really excellent-looking program. A few reminders: if you have a question after a presentation, just raise your hand, and we'll bring the microphone to you. If you just wait until you get the microphone, because people just cannot hear otherwise. If you have questions not related to the science of the presentation, please ask Liz and Christina Donna, who are at the table just outside this meeting room. Again, please do talk with each other and with the sponsors during the coffee breaks and at all other opportunities. There's a specific NMSG Wi-Fi access code. The network name is Neuromuscular Study, and the password is up there, so that's good.
All right, okay, my turn. Well, if you have not done it already, become an thank you for advancing the slide, Gil. Become an NMSG member. So, you go to the NMSG website, and you apply. The executive committee does review your app. It's a site approval. You apply as a site, and the cost is $500 annually, and it covers every researcher at your site, or every clinician/researcher. So if you're a 2-site person, $500. If you're a 50-site person like Hannah, $500. You got a good deal. So yeah. And so if you have any questions about that, please again talk to Liz or Donna. Okay, you're next.
Okay, so here are the benefits of being an NMSG member. Of course, the meeting is the highlight of the year and the interactions that we achieve at the meeting. But stuff happens in between the meetings that you can take advantage of. So there's a discounted registration for the meeting as a member, but there's opportunities if you contact us for brainstorming potential study ideas and helping develop a steering committee for a study. We're also very happy to try and support critiquing grant submissions. There's quite a strong history, actually, which we really prioritize, of supporting younger investigators through mentorship. There are opportunities around database management and statistical support, principally through our links with Rochester. We have a new opportunity, which is, Rick, you may want to talk a bit more about this, for modest funding assistance, where principally with junior investigators, if they have achieved a grant, for example, a small grant, but need some additional support for certain aspects of it, we may be able to offer some modest support. That's a new development which you may want to comment on, Rick. Then, of course, there's the Shark Tank competition, which is a highlight of the meeting. It's really a nice event. Young investigators presenting their ideas and then critiqued by a group of sharks. And I think this year, there's also the audience can vote on the presentation that they prefer. And of course, you get to hang out in cool places like Italy and even sometimes England. Yeah.
No, the last one's the best, isn't it? Hang out in cool places together. So there's a lot we can riff on this one, and there may be questions you all have that you can come up to us later. As you develop new studies, we want you to consider making them official NMSG studies. And there's a process for that. It's on the website. If it becomes an official NMSG study, then we help you form a steering committee. Mike, the Rochester team can provide preliminary statistical support that helps you develop the statistics for the study. It doesn't do the statistics, but it helps you develop the statistics. They've been doing that for us for 25 years, Mike McDermott and his new successor, Dr. Wahid. So that's great. And of course, if you become an official NMSG study, then we'll help you critique the grant submission. Then if your grant gets funded, if you use Rochester, then we want you to put them in as the person who are going to run the stats, so then there should be money built in the grant to do the stats and the database. If, though, you're not required to use the Rochester team, and if you get a grant funded, it wasn't an NMSG study, but you're an MSG member, and you actually need a little bit of money for some extra support, if it was a foundation grant, you didn't have a lot in there for statistics or data management, there is a form on the website where you can apply to up to $10,000. We've actually only had one person do that in the last year or so. Dr. Kelly, Chris Kelly, did it for one of the posters that she showed last night. And we supplied the funds, then she had a statistician she used. Or you could use the Rochesters to do the stats and pay them for that. So that's the idea. So we hope more people take advantage of that.
Slide is still you, I think.
Oh, it's still me. All right, sorry, maybe that was too long on that slide. This is the NMSG member sites, and look at it for a second. That's not the NMSG member sites. That's our platinum sponsors. Thank you. So look at that for a second. If you don't see your name on the list, talk to me or Mike or Liz or Donna. You should be. I think in the past, sometimes I would highlight who is late on paying dues. It was always Amato. I think you finally paid up, Amato, finally. But we didn't do that this year. But please make sure your name's on the list, and if it's not, we'll get it on the list, and if you're not an official member, we'll get you to be a member.
So once again, it's really important that we thank all these colleagues who have really worked hard to make this meeting such a great event. So they're all listed here. I'm sure you'll say thank you to the ones you know personally, but maybe if we just collectively give them all a big round of applause. Thank you to all of them.
I think we highlight them again on the next page, right? We start. Those are thanks to. Now we have sponsors. All right. Okay. Sorry. So once again, we just want to remind everyone who our sponsors are. So at the platinum level, we have Avidity and Johnson & Johnson. I know I've talked to a lot of Johnson & Johnson people the last day. Haven't talked to Avidity people, so please say hello.
And then, again, just to emphasize how much we value the partnership with all our colleagues in industry. So the gold level sponsors, Alexion, Arginex, Catalyst, Merck, Sanofi, Sarepta, Scholar Rock, and then silver level Cartesian, Immunovant, and NHTI. So thank you to all of them.
I talked to the Scholar Rock people yesterday. That's the coolest name. Yeah. So that was good. Your PR people did good on that one, Scholar Rock. All right, and then, is there one more? Oh. No, we did that. And then bronze. So bronze is Dyne Therapeutics, NMD Pharma, Solid Bioscience, ML Bio Amgen, Caverna Therapeutics, and Octapharma, and then also we've gotten support from Loop and Neuroscience and the GBS-CIDP Foundation. This year we have different sessions sponsored by we give industry the opportunity to sponsor different sessions. So UCB, is sponsoring the Shark Tank competition tomorrow. The Young Investigators session that was yesterday, Arthex Biotech, and then the poster session that was yesterday was sponsored by PepGen Bio. So thank you. Is that, yeah, maybe then, yes. Before we go to the final slide, Gil, can you just turn the slides off for just a second? So I thought we'd just take a moment. In the last several months, we had 3 giants in neuromuscular disease pass after long, amazing careers. And I just wanted to acknowledge that. We all, as Isaac Newton said, our success is based because we stand on the shoulders of giants. And these are 3 giants. Austin Sumner died a few months ago, a giant in neuromuscular disease, first in Philly and then a long career in New Orleans, and an incredible peripheral nerve person and electromyographer, really advanced the electromyography field incredibly. Dr. Julaine Florence, a doctor of physical therapy, she passed. She'd been to this conference many times in the Beaver holidays. See Emma nodding. We all remember fondly Julaine. Julaine was such a pioneer in the field of physical therapy, and she was part of the Griggs, Mendel, Fenichel, Brooks CDD group, and all those neurology giants had Julaine or Shri or Wendy behind them, making that original Duchenne study, a natural history study, possible, and then the multiple drug trials. And as I was going through the posters last night, and I was seeing some of the amazing Duchenne gene therapy and some of the natural history studies that are still collecting, I was realizing that all of that would never have happened without the CDD group and people like Julaine Florence. And then finally, Dr. Peter Dick, an incredible giant in the field of neuromuscular disease and specifically neuropathy. Peter Dick founded the field of modern peripheral neuropathy. So everything we do in peripheral neuropathy in some way relates back to his work at Mayo Clinic over a generation, and we're so blessed to have his son Peter Dick, Jim Dick here. Where are you, Jim? Jim Dick here, as which is probably one of the most amazing legacies that Peter has given us. So let's just take about a few seconds to have a moment of silence for all 3 of those great giants. All right, thank you very much. And now to finish.
Okay, yeah. So the highlights for today, there's a number of really, I think, excellent sessions ahead of us. So AI and E in health, regulatory requirements, new clinical phenotypes, industry updates. And then we have the NMSG Fellow presentation this evening, which we're all looking forward to. And then, of course, the Robert C. Griggs keynote lecture, which will be delivered by Valeria Sansoni, which we're also very excited about. And then, for those of you that have registered for the gala dinner, which is actually sold out, you need to assemble at 6:45 to get a short bus ride, I think 7 minutes up the hill. We're hoping we'll get there before or during sunset, it depends on the weather. There's a fabulous view from this villa perched on the top of the mountain, which hopefully will be a really nice evening. Then just finally, I just wanted to make a couple of comments about the poster session, which I think was fantastic. It was a great atmosphere in the room, some fabulous posters, so well done to all of the colleagues who did posters, and particularly to the 2 winners. And we just wanted to make it really clear that we have quite a rigorous process of selection and prioritization of the posters and how we select which are platforms and also who is funded. And it's based on merit as judged by a panel of poster judges. So it is not the case that any poster will get funded, but obviously the standard is high. So I just thought Valeria might want to come up and just give a bit more sort of, with Emma, actual facts about that process so everybody is really clear.
Yes, just a few seconds. So just again to make it clear that it's really a positive thing that NMSG is supporting this. So 165 abstracts were submitted, 144 were accepted to be presented, Of these, there were 5 platforms selected and 5 flash presentations, and 86 people of the abstracts submitted were completely funded by NMSG, including then, in addition, the Shark Tank presenters, the last Shark Tank winner, and the executive committee, the planning committee, and all the invited speakers. And the final thing I just want to say that the abstracts had a scoring system, so there was a grid based on from a 0 to 5 on significance, clarity, and impact, and it was distributed between 3 senior people in here. So just to give value to the ones selected, and also to think that we are trying to raise the level, and I think it's a very good thing and quite unique that NMSG is doing. So it's not given for granted, and I just wanted to give value to what we are all doing.
And I just want to quickly highlight for obviously all members, but especially for the new members or the new people or early investigator, to please stop by. There is a desk beside Liz's desk about our data coordination center in Rochester. And like Rick said, we have a very long-standing relationship with the MSG. And our data center manage, study design and system programming, reporting, and data management. And one of the advantages is also our statistician and the long history with the MSG group, and also quite competitive fees compared with other CROs. So please, if you have time, stop by the desk, learn about the data center. And there is a web address where you can read more the details of the services offered on the NMSG website. So thank you.
Thanks, Emma. Yeah, I'm glad you expanded on that. That's great. The picture yesterday, Liz sent me a copy of the picture we took. That was a great picture, very unique. So most of us got on there. A few of you had already drifted away. But Gil, tomorrow, can we put the picture up maybe in the morning? And then you'll get it all sent out and it'll be on the website. So it's great.
So just to the points that Valeria made, I think it just emphasizes the commitment NMSG has to young investigators to support them, but also to support a high standard of presentation. And I think we should give Valeria and all the poster judges a round of applause for a huge amount of work that goes on. Great, so thank you. So we're going to go to the first session now, and the moderators are Professor Eugenio Mercuri and Dr. Nicholas Stryker. Thank you.
Well, I'm glad everyone has arrived now after yesterday night's nice dinner. So welcome to the first session, which is on AI and e-health. And we have an interesting panel covering different topics. The first speaker is John Thornton, and the talk is about MRI as a biomarker in neuromuscular disorders. Dr. Thornton, please.
Thank you very much, and thank you to the organisers for inviting me to give this talk. I'm a magnetic resonance physicist by trade, but I'm presenting very much on behalf of my colleagues at University College London. I just have one disclosure. So, for the purposes of this talk, I've divided quantitative biomarkers into 3 categories. The first is what I've called the direct MRI biomarkers, where we're using MRI to measure quantities which have somewhat obvious interpretations. So this would include muscle cross-sectional area and muscle fat content, where MRI is the means of measuring them, but they're not MRI quantities. And these are specific, easily defined, and easily understood indices of disease. Then we can consider what I've called physics-based MRI biomarkers, which are really intrinsic to the MRI measurement process. So these require a conceptual or a physics-based model to relate the findings to the underlying pathological substrates. And the example I'm going to discuss today is T2 mapping, but there are many others. And then there's a category which I've defined as being dynamic MRI biomarkers, where we can use MRI during an active process while the participant's in the scanner to access metabolic and physiological measures. And the example I'm going to discuss in this talk is phosphorus magnetic resonance spectroscopy. So I'm going to frame things around those three categories. I'll start with the method which is probably familiar to most people here, which is Dixon chemical shift-based fat water imaging, known as Dixon water imaging, in which we typically collect a number of typically 3 gradient echo images with successively increasing echo times. And because water and lipid hydrogen protons resonate at different frequencies, their signals go in and out of phase as we increase the time from when we excite the system. And because of the phase difference in the images, it's possible in a fairly straightforward way to derive images where we separate the water and the fat proton density contributions. So we have an image which is proportional to the water proton density and an image which is proportional to the fat proton density. And given these two images, we can then calculate what we call the proton density fat fraction. It's proton density weighted if we've been careful with our experimental design to avoid other weightings. And then if we perform an anatomical segmentation of the image, we can extract as outputs per muscle fat fraction, typically expressed as the mean fat fraction, the fat content, and also the cross-sectional area of the muscles. So we get these two sort of fundamental quantities in neuromuscular MRI. We can, of course, access cross-sectional area using simpler MR techniques, but in our practice, this is how we've done it. Then there have been many studies over the last 10 to 15 years from our centre and many others, including colleagues here, that have shown that muscle fat fraction is a responsive measure of biological change in progressive neuromuscular diseases. So this is an example from our first major published study in Charcot-Marie-Tooth disease type 1A, where this plot shows the change over 12 months, and we were able to detect in individual patients changes in muscle fat fraction content of just a couple of % over this time scale. And this means that when we look at this in terms of biomarker responsiveness, which we express in terms of the standard response mean, which is the mean change divided by the standard deviation of the mean change of that measurand, that the calf level MRI fat fraction change over 12 months in this particular group of patients, the standard response mean for this measurement greatly exceeded all of the clinical measures and also the calf myometry, illustrating the potential value of quantitative MRI biomarkers to enable treatment trials with small or realistic numbers in rare diseases. So this was something we published almost 9 years ago. Just this year, Matthew Evans was the first author on our paper where we presented the 4-year follow-up data from this study. And the key points here first are that stratification by baseline fat fraction increases the 12-month responsiveness of the fat fraction change. So comparing the standard response mean of all of the participants compared with if we stratify them according to their baseline fat fraction, we get a much more sensitive marker. So that is one finding from this paper and other studies that we've done. The second very important observation from in this paper is that the fat fraction change at 12 months predicted the clinical changes that were measured up to 4 years. So the MRI probably has validity as a bridging biomarker. because the clinical ratings didn't change significantly at 12 months. We've also looked at calf level muscle MRI measurements in other types of CMT, where we've used the same protocol in participants with CMT-X1, CMT-1B, and CMT-2A, and followed them up at approximately 12 months. The follow-up intervals were increased due to the COVID pandemic. And what we saw in these other groups of CMT, similar changes to those we see in CMT-1A, with some difference relating to the different phenotypes of those three subgroups. The potential validity of the biomarker was again confirmed by comparison of the calf muscle fat fraction measurements with, for instance, the CMT-ES score. And again, the change over the interval of the study, the standard response mean for individuals with muscle fat fractions between 10 and 70% was 1.2, which is quite similar to our previous study, supporting that the MRI biomarker may be useful across all of these variants of CMT, which I understand covers around 90% of the potential trial participants. In order to increase the number of potential trial participants, particularly towards the younger age groups, under the leadership of Mary Riley, we, and in collaboration with our colleagues in the University of Iowa, undertook a study looking at the foot muscle in patients with CMT1A in much younger patients with an age range of 6 to 20 years. And by looking, by imaging the foot and performing our fat fraction measurements in the foot, we were able to achieve statistically significant changes in the foot muscles that weren't evident in the calf muscles because of the length dependency of the condition, which was a nice increase to the potential pool of trial participants. Another area where we've done similar studies, looking at these, what I've termed as direct MRI biomarkers, is amyotrophic lateral sclerosis, where the disease progression, of course, is much more rapid than in an inherited neuropathy like CMT. And we found in this study that, in fact, as you might expect, it's the muscle cross-sectional area that was the key readout. And we showed change over 6 to 12 months and also satisfying correlations with functional measures such as plantar flexion strength change, comparing it with the remaining muscle cross-sectional area of the respective muscles. We've also moved beyond measuring just lower limb, and for this group of patients with ALS, we found the hand imaging to be very sensitive. So we adapted our protocol and our segmentation method to be specific to the hand. And what we showed was that the hand muscle cross-sectional area measurements were actually more responsive in terms of their standard response mean than the thigh and calf level muscle measurements we made in this patient group. Supporting the idea that for future studies in ALS, it will be important to address probably the whole body using the latest MRI acceleration techniques. So MRI fat fraction and muscle atrophy, because fatty atrophy is a common pathway for many neuromuscular disorders, is becoming an established secondary outcome measure And it's been explored now in many conditions. Those listed here are those that I've been involved with in my time at Queen Square, but of course other colleagues here and internationally have looked at many other diseases. The method is readily deployed on standard MRI systems. It's compared with other MRI techniques, robust to minor implementation differences, so easy to replicate between scanners and sensors. And we are working on applying the new improvements in MRI technology to accelerate the acquisitions and increase the anatomical coverage. So if we move on, as part of this process, we've developed an automated segmentation technique, which is where the AI comes into this talk. Previously, extracting the MRI biomarkers was labor-intensive and time-consuming. It involved manually delineating the muscle boundaries on the images. Now we can use a convolutional neural network to do this, which has been trained on our existing research data. And we've been able to show in the first iteration of our first version of our software, which was developed by Baris Kamba, our colleague in UCL Computer Science, that the automated method is highly effective. It just requires some quite minor manual checking and correction. But even without that, we managed to replicate the manual segmentation results very successfully. And this is a real advance in the tools we have to perform these sorts of studies. Since we originally published our muscle sense segmentation algorithm, Dr. Kamba has improved it, and it now works. It's able to segment individual muscle compartments and can use as inputs more types of MR contrast than the 3-point Dixon imaging. And also, we're now extending it to work in other regions, including automated segmentation of the hand. So this is a vitally enabling tool to the use of MRI biomarkers in neuromuscular conditions. So that was covering the important area of muscle fat fraction and muscle area, which are already finding use in treatment trials. I'll now move on to what I've called the physics-based biomarkers, and the example I'm going to give is muscle T2 relaxation time. So T2 is a measure of how quickly the MRI signal decays after the first excitation in the scanner. So we collect a series of images with successive delays from the excitation, and in the simplest case, we see an exponential signal decay. And we can fit this decay for each pixel and generate a quantitative T2 map. And this T2 relaxation time constant depends on the distribution of molecular rotational frequencies within each voxel, each pixel at volume. And this changes with tissue hydration or edema, also sensitive to microstructural changes. And if we don't perform our experiment carefully, it also can be dominated by changes in lipid content. So our hypothesis was that muscle water T2 changes are probably dynamic and are probably sensitive to earlier changes before the fatty atrophy occurs, which makes them potentially attractive as potential biomarkers. However, it's complicated to do. The values you get from the analysis, unless you're very careful, can be highly dependent on the implementation. There's a confounding signal from fat with long T2. And we have to deal with the fact that the noise distribution in MR images is non-Gaussian. So we, in common with a number of other centers, have developed an extended phase graph model, which is basically a physics model to accurately model the decay in these sequences, which in the real world is not truly exponential. And we model the fat and water signals independently and fit them to the data so that we can extract a T2 map, which is just from the muscle water independent of fat. And this works reasonably well. In our implementation, it's computationally expensive. It takes a long time, but we can do this. And for instance, here you see in a healthy volunteer, the muscle T2 is a uniform value of around 30 milliseconds at 3 Tesla. And in this particular patient, the T2 was elevated in various regions, and those regions of elevation of T2 are different to the elevations of muscle fat fraction elevation. So we've started to explore what the potential value of this measure in a number of our studies. So this is data which is from a subset of the participants in the study. I presented a few slides back where we've looked at calf level muscle T2 changes in these 3 variants of CMT. And you can see in the left-hand column that compared with the controls, there was regional T2 elevation in these example patients, which was reflected in the baseline values, where in the patients there was a distribution of higher values compared with the controls. This okay, I'll move on. And we've seen similar T2 changes in ALS, with water increasing significantly over time. But in this case, the changes were less responsive than measurements of cross-sectional area, and the evolution of the T2 and the CSI and the cross-sectional area changes were different. So T2 is a potential biomarker, but it can decrease as well as increase. This is an example from one of our papers where at end-stage inclusion body myositis, there's a decrease. And more work is needed to understand these changes and also to improve the computational efficiency and be able to transfer the method between systems. The final method I'll describe is a dynamic MRI biomarker, which is putting the scanner into a different mode to perform phosphorus magnetic resonance spectroscopy, which allows us to directly access high-energy phosphorus metabolites, which obviously are key to muscle energy generation. And by performing an exercise paradigm within the scanner, we can detect the change in phosphocreatine that occurs during exercise and then track its recovery afterwards. And our interest here is in mitochondrial disease. So with our colleagues Chiara Pizzamiglio and Robert Keithley, we set up a paradigm where we're performing exercise in the scanner and measuring the recovery of phosphocreatine. And the early results are very exciting. The phosphocreatine recovery time constant is significantly increased in patients with primary mitochondrial myopathy, and more so than the baseline changes in phosphocreatine. And there's good correlations with the clinical readouts suggesting this is a useful potential biomarker in mitochondrial myopathy. So, very finally, having developed these methods in common with other international groups for group-level measurements in treatment trials, we also want to be able to apply these methods for individual patients in radiology. So this involves taking the patient's images offline to generate a quantitative radiology report, which can then be transferred back into the hospital PACS system to inform the reporting radiologist's report. There are commercial solutions now emerging for neuro applications such as dementia, MS, and epilepsy, and we're very keen to see whether we can achieve the same thing for patients with neuromuscular conditions. So we have a project underway to do this, and it involves the the automated segmentation I've described, and similar sequences, and generating a graphical report, which is presented to the reporting radiologist on their PACS workstation to reduce inter-reader variability, decrease uncertainty for visually equivocal cases, and enable a contextualization of an individual's quantitative MR readouts versus a reference population. And we have a prototype report that we're now testing in a systematic study. So to conclude, there are these I've coined these 3 categories of muscle MRI biomarkers. The direct MRI biomarkers, muscle cross-sectional area and muscle fat content, are ready to use. They're robust, valid, and responsive in many diseases. We are now awaiting data showing sensitivity to treatments, and there are a number of studies coming to an end, and we're expecting some exciting publications in that area. And as I mentioned in my last two slides, this is close to being ready for radiology application to benefit individual patients. The physics-based MRI biomarkers are more complex and more work is required. And for certain niche clinical applications, there are some exciting emerging dynamic MRI biomarkers that reflect metabolic and physiological function. I'd like to acknowledge all my co-workers and all of the volunteer participants in these studies. Thank you.
We started a bit late, so we can allow two very quick questions. There is one hand there.
As we design clinical trials that may run a year, this is effective, but in this age of rapid weight loss and GLP-1 receptor agonists, do you think that that would have impact on clinical trials and outcomes that are MRI-based?
I'm sorry, I didn't catch that.
So if people lose 20% of their body mass that's fat,
Okay.
will that impact your studies and outcomes in clinical trials?
I don't think so. Obviously, those statistics will be collected and statistical correction can be done for that. It's an interesting question of whether that would be a fundamental confounder. Maybe we can talk about it afterwards. But yeah.
Thank you.
Okay, next up we have AI in healthcare. How can it help and where do we stand? So we'll bring up Salverio DiMarco from Humanitas Hospital in Milan.
Good morning, everybody, and thank you so much for inviting me here. Such an honor to be here. So my background is I'm a biomedical engineer and working in the AI field since 2016. So with some years of experience in trying to apply AI. Mostly of my experience is in the field of oncology, but recently in the past years I'm trying to apply also into the clinical practice some AI tools in neurology field. So I'm very glad to be here and try to point at the table some point of discussion about the application of AI. So with my intervention of today, I want to, let's say, highlight artificial intelligence in healthcare, and in particular in trying to ask the question, how can it help and where do we stand? To answer this question, I would like to give you a practical example of how we are actually using AI and how we actually build AI and then validate it. First of all, I'm going to give you a very brief introduction just to have a common vocabulary, common diction to everybody, just to understanding better all of the concept, more or less high level, not so technical, I will promise, in this slide. So first of all, AI, it's a very high and complex field of applications with different concepts concatenating each other. You can think AI as, let's say, a matryoshka with different concepts behind that. Of course, inside artificial intelligence, we can find machine learning, a very familiar term. I think everybody more or less knows about machine learning, but in a few words, it's some mathematically, let's say, functions and so on that are trying to understand patterns from historical data and then trying to use these patterns in perspective data to answer such questions. Then we have the deep learning, going more deeply. Deep learning is just machine learning, but with a I would say, powerful tool, which is the deep neural network that I'm trying to power up machine learning and use this problem to solve it, to understand more complex data like images, video, signal, and so on. Then we have a last, let's say, innovative branch of AI, which is computer vision, of course, but then generative AI. The generative AI, which is maybe, you know, of course, which is the technology behind some powerful tool like ChatGPT or Gemini or other large language model, but it's just a lot of deep learning and deep neural network architecture trained on a lot of data, more or less all of the knowledge about the internet. And in general, once we understand, let's say, the linked concept about artificial intelligence, we can better understand the application of AI in healthcare. And as you can see, talking about AI in healthcare is a general purpose, is a general application. AI in healthcare means medical product, medical device, software, and decision support system. But once we go more deeply, we find that once we understand more deeply the technologies, we can highlight, we can define more in a more precise way all of the single and specific application we can do. For example, with generative AI, which is our last branch, our powerful tool from AI, we can do this new innovative approach called synthetic data generation, or we can do preventive care, or we can make this approach called a digital twin for, let's say, help the clinicians in understanding better the behavior of the patients. In general, AI is divided in these two main branches, discriminative models and generative models. Discriminative one are, I would say, the more traditional ones. So taking some data of input, we can understand the input, we can understand the pattern, then extract information, extract feature, create a prediction, and something like that. With generative model, we change the paradigm. So we have a prompt, we have some input data, and then we can create evidence. We can generate data, we can generate images, and so on. Generative AI right now is one of the most powerful, let's say, tools and the most powerful things about AI that we have right now. But of course, generative AI is a very high complex and high level approach. Generative AI, it is the technology behind ChatGPT, but it doesn't mean that generative AI is ChatGPT. As you can see, I don't know if the pointer works. Yeah, as you can see, ChatGPT or Copilot are just a very small point in this overall information about generative AI. Generative AI means a lot of tools and a lot of algorithms, and right now it is the state of the art of the artificial intelligence model, not because we have this kind of application, but because the technology are evolving, and of course also the algorithms are evolving. And right now the state of the art told us that generative AI approach means better performance above the model. So once we defined what is AI and which is the kind of technology behind AI, we can use AI to answer clinical questions. So in my perspective as an engineer, as someone that trying to use AI to solve problems, AI is just a technologies, and we are trying to use technologies to solve a clinical problem and clinical unmet need. Also, everything starts about the data. Also, the European Commission told us that the data are so important, everybody knows about that, and the European Commission trying to create a lot of, let's say, consortium, a lot of initiative due to the sharing of the data and the privacy preserving sharing of data with European health data space, data governance, and so on. So everybody agrees that we need data to better research, to do better research, and to understanding clinical need, and of course to accelerate clinical innovation. How AI can help in solve this problem? So AI is like a technical tool, technical measures that we can use to answer and solve this problem, particularly the availability of the data. Some of the technologies that, for example, the European Commission identifies as, let's say, solutions to this privacy-related problem in terms of sharing data are federated learning and synthetic data, which is one of the two main applications I'm going to explain you this morning. Of course, when we talk about AI in healthcare, it's a very complex field, it's a very complex application. We cannot talk about AI like we talk in other, let's say, domain like banking, insurance, and so on. Everybody knows, of course, that this is something we really know, but I would like to highlight this concept. So using AI and validating AI is not just using a tool, but it's a matter of understanding the model, create model that needs to be transparent, validate model with an external code of validation and so on, and then the model needs to have, at least in the European country, of course, but need to taking care of the protection of the data and the data sharing. That's this probably from our work, but that's our main, let's say, workflow of application. We are trying to use technologies and expertise from a European Commission, so very big, large consortium of application, consortium of partners, from extracting clinical information, knowledge, and data in this privacy-preserving way. And then in my institution, we are trying to create very specific and low-level and focus AI department, trying to use AI, understanding AI for creating such application to solve the clinical problem I mentioned before. So the core of this presentation, I would say, is the identifies how do we stand like the AI application, and I identified 6 main, let's say, clinical application or practical clinical application of AI nowadays, in particularly from our work, but in general the work of the European consortia in which we are in. So starting from natural language processing system, trying to identify what is the problem and which is the problem. The problem, of course, is the data availability. Right now, these days, this data is so critical. So the 97% of the data within the hospital are in unstructured format. That means that 97% of the data of the hospital remain unused for clinical purposes. So how we can use AI to solve this problem? So to answer this question, we are trying to understand, we are trying to ask each other what is the, let's say, the source of the data. Also for producing a medical content. And the real source of the data in the healthcare application, in the healthcare field, is right now the voice. So every kind of data starts from the interaction between patient and clinician, between the patient and the clinician through a conversation. So why we can use the voice interaction, the conversation, to automatically extract information and then automatically extract into a structured database, a table, and so on. So we created, and right now this is under, let's say, clinical trial, clinical study with real patients within our institution. We are trying to create this voice platform called Ambient AI to automatically extract information through the conversation between patient and clinicians. Of course, this isn't just a transcriber, but is a more complex platform that, starting from the interaction through the voice between patient and clinicians, are going to create, of course, the medical text report, but then extract information from the medical text report and then automatically put this information into structured electronic health record system. Moreover, the electronic health record system is connected to, let's say, a database of clinical protocols, clinical trial, and then highlight in real time to the doctor some clinical trial or clinical study in which the patient, for example, is suitable for. I already like this concept, but once we have the transcription from voice or something like that, we can automatically instruct structured information to solve the problem of the 97% of unstructured data. Moreover, think about this kind of application, this voice model, also for, let's say, patients remote monitoring, and in particular, to use a voice, to use the phone just to call the patients with an AI agent and asking the patients what is their, let's say, adherence to a therapy, or what is some of this information. So this is something that we are trying to, let's say, use nowadays. Also, this application is nowadays in clinical trial application, and this is an application for calling the patient and instruct for remote patients monitoring. Another very fascinating from my side, but also very innovative application is the generation of synthetic data. Synthetic data is another application of generative AI in the healthcare field, and is basically the concept of generating artificially data that are mimicking real-world correlation in the real data. So synthetic data are just artificially data that are mimicking a relationship produced by an AI model. The main application and the main innovative concept behind synthetic data is that, first of all, synthetic data are totally anonymized data set. So synthetic data also for the European Commission, also for the European AI Act, is considered anonymous data. So we can use synthetic data to avoid, let's say, classic anonymization process and to sharing data in a privacy-preserving way. And second thing, synthetic data are, let's say, conditional data. So we can ask the model to produce whatever we want data with the characteristics we want. For example, I can ask the model to generate hundreds of patients with specific characteristics, specific comorbidities, and specific, let's say, response to treatment. The process of generating synthetic data is I would say not so complex, but everything, of course, starts from the data. Basically, the process is divided into three main phases. The most important one is the phase one, because we need to understand the complexity of the healthcare data, and we need to choose the right model to, let's say, address all of the clinical aspect behind the data and address the clinical questions behind this data. Then we need to train the model, of course, and then we can use the model, as I said, to generate whatever we want data. One of the most important and most innovative and fascinating, of course, application of synthetic data is, of course, the clinical trial. Everybody in this room knows about the complexity of clinical trial, and in particular the complexity of a randomized controlled trial, but think just for a second, if we can use synthetic data to produce, of course, in a preliminary way, so for preliminary result, synthetic control arms, in order to understand better, let's say, the results of such trial. This is already something that we are actually trying to build, and with that we are actually already trying with some trial in the oncology field using, let's say, some application on platform that we build. One thing that I want to stress is that the focus of my AI center in the hospital, so in general of my work, is not to use AI tool, use AI platform like Google or ChatGPT or Gemini. That's why European Commission, and particularly also Italy, provide us to use such this tool due to privacy preserving aspect. So we are trying to build our own model and to locally deploy within the hospital, so also to help the hospital in having a practical way such this technology. Talking better about synthetic data, also with behind the generation of the data, the most important part is the validation part. So that's why we are also creating a synthetic validation, the so-called synthetic validation framework, in order to validate with three specific metrics let's say, the level of accuracy of the synthetic data with respect to the real one. The framework built, let's say, on the basis of the World Health Organization guidelines about AI, measure the quality of synthetic data in three metrics, which is synthetic fidelity, clinical utility, and the most important one, privacy preserving, because we need to know that we are not copying real patients, but we are, let's say, imagining generating brand new one. We already have some experiments, we already have some results, of course, in clinical trial. In the oncohematological field, we are actually, we replicating a clinical trial, a retrospective clinical trial, using a synthetic control arm generated from a retrospective cohort, and we gained all of the clinical endpoint, and of course, we published this clinical trial just to, let's say, highlight the validation of this study. Also, we have another example in the field of neurology, and in particular in multiple sclerosis. We use synthetic data to replicate two different harms without missing data. So basically, the problem of this study is that we have more or less 5,000 of patients, but with a lot of missing information. So to solve this clinical problem, we use synthetic data technology, starting from the more or less 1,500 multiple sclerosis patients, but with all of the data, without any missing information, also with the MRI information, and trying to use this cohort of patients to train a generative model, and then ask to the model to generate the original number of patients of the original study, replicating, of course, all of the statistical and clinical characteristics, but moreover, using the synthetic data to build a prognostic model, a Cox model, and a prognostic model, and identify that using synthetic data we have more results with respect to the real one. As I said, synthetic data is considered anonymous, not from my side, but from the AI Act. But what about the status of the regulation about clinical trial using synthetic data? In the US, we have only 3 cases of drugs that gain the approval using synthetic data, and also we have, let's say, an alliance between the FDA and the scientific community in using synthetic data for the most critical concept. In Europe, we have, as I said, different consortia, so different partnership between research center, hospital, and industry in using synthetic data for clinical control arm. The most important one is Cynthia, which with the aim of applying this technology in different medical fields. In conclusion, I would like briefly to present federated learning. Federated learning is a new way of training the AI model, so for a new paradigm of training. Instead of sharing data, we are trying to sharing the knowledge of each artificial intelligence model in different healthcare hospital. One example of federated learning application going from this European consortia GenoMed for All in the field of hematology. As you can see from these graphics, this is an example of 3 different hospitals building each other a machine learning model for predicting the overall survival of this hematological disease. Of course, the worst performance is from the hospital with the less amount of data. But once we use federated learning without sharing data, with just sharing the value of the model, the knowledge of the single model, we can reach the same performance of the centralized one. Right now, this is a really practical example because we deployed this solution actually in a different node of the hospital, and right now this is, let's say, active in the European Commission, and we are going to use a federated learning approach to enhance the performance of our model. In conclusion, I would like to present the concept of the digital twin, because all of these AI technologies can lead and can bring all together into just one concept, which is the digital twin. Digital twin, in our perspective, is, let's say, a way to build a clinical decision support system that works on the individual patients and to provide clinicians a way to guide the decision in taking, for example, for diagnosis, prognosis, and therapy optimization. Not only to ask clinical trial, as I described with synthetic data, but also to help clinicians in taking their decision. Of course, building the concept of the digital twin is a very complex thing, and it is a very complex application, not only in building technology, but also in validating technology. We have a lot of issues. More of these issues are related to the data availability. So, in conclusion, all of the technology that AI, artificial intelligence, can bring us nowadays can be used to solve this clinical problem and to practically lead to the building of a digital twin. For example, with synthetic data, federated learning, a large language model, we can solve the data availability problem in a privacy-preserving way, and we can, in conclusion, build the concept of the digital twin, which is a way to digitalizing and integrating such different data modalities in order to create a copy of the patient automatically explainable and interpretable by the clinicians. Thank you so much.
Thank you so much. Definitely, this is the future. I think we have a minute or two for questions, if we have any from the audience. One question back here. I think we're coming.
Thank you for this exciting presentation. When it comes to the bedside, clearly AI has a role in generating the history component, and it's kind of malleable. When it comes to interpreting the components of the history, there's different weights, different components of history, elements. So that to get to the next step, which is integrating this into an intelligent impression, diagnosis, plan of care, which may be the next step, next challenge, I think AI has some shortcomings in being able to assign different levels of importance to different symptoms that are being spitted out by patients. How do you see that being resolved in the future, and how soon?
Exactly, very interesting point, absolutely. So basically, I think that once again, the technologies can help also in this field, and in particular AI. There is some kind of AI application that are going to answer directly to this question. For example, we can use AI to identify which is the most important data and symptoms of clinical evidence related to a disease or related to evolution of some disease. Of course, we really need to build a tailored model about a disease. We cannot use generalistic approach. So basically, we cannot use generalistic AI to answer this important question. We need to build a different AI model tailored for these questions, and also we need to build specific model to explain the results of ChatGPT's model in order to, let's say, give the clinicians, give the human at least, all of the evidence to understanding if AI comes into the right decision, and if the, let's say, highlighted information from the AI are going in the right clinical directions. So AI can solve this problem and can address this issue, but a tailored one AI. So not a generalistic approach. We need to build specific AI model for understanding the most clinical evidence and clinical important feature from a single patient.
Okay, thanks so much.
Thank you again.
And we go to the last talk in this session, which is clinical trials powered by AI by Dr. Ritter from Sanofi. Dr. Ritter, please try to stick to your 15 minutes, otherwise there is no time for discussion, and it's a shame because this is all very interesting.
Will do, certainly, and Severio took a lot of the hard work off this, so this will be good. So everyone knows. So my background is in surgery and bioengineering. So I'm going to be less of the scientist and more of the engineer. So just remember that when you think of the way I look at stuff. So we're going to talk about it. Kind of Severio covered the basics of how AI works and kind of the capabilities in healthcare. So if we go to the next slide, we'll talk specifically about how it works in clinical trials. So we're using it in a bunch of different places in clinical research. So on the first slide, we kind of go through what those kind of capabilities are. So when you think about it in terms of trial design through the kind of setup and patient finding and enrollment phase, through operations and kind of the execution of the trial itself, data collection, and then kind of review and analysis. So exactly the parts of a clinical trial that you would expect and kind of exactly the way you'd expect it to work. So very much in the design phase, using some of those concepts that we talked about in the last 15 minutes with digital twins and with disease modeling, being able to look at kind of the optimization of the design of the trial, the optimization of the endpoints, understand patient dynamics and disease dynamics to kind of fully understand what it is that you're looking for in terms of medical evidence. So we've done kind of extensive work with this, both internally and with a lot of academic partners, to be able to optimize the way we design the research for the drugs. The next one, which we'll talk a little bit more in a second, is really about kind of finding the right patients and then using some other AI techniques throughout the rest of the process. So some of these are kind of in the medical sphere, so looking at kind of digital twins and patient finding and things like that. And then some of them are very much kind of AI for the operational aspects of like trials are a pain in the ass. And so Gen AI for documentation, being able to do other sorts of automations, drug supply automation, other things, just to smooth out the lift of running clinical research. And so that's a big part of this, especially for my team, is making sure that not only are we progressing kind of the science of the diseases that we research, but also progressing and improving the burden that falls on healthcare practitioners in terms of the execution of clinical research. So there's a big kind of component of that you'll see throughout these, especially in the operations categories and in some of the other factors, looking at drug supply, looking at kind of operational performance, deviation prevention, training, things like that. Being able to streamline some of those things, make it easier for people to participate, make it easier, especially many people that I've talked to here are from academic centers, have big staffs and support. As we work with smaller centers or individual HCPs, it becomes very challenging to participate in clinical research, which then undermines the ability for us to offer clinical research as a treatment modality, because if it's only limited to the academic centers, that's a challenge in some areas, especially in rare disease and other indications where that's problematic. So really trying to kind of make it more available for people. Then when you get into the data collection space, so the two I'll spend time on today are kind of the patient finding and the data collection. So when you get into the data collection space, starting to use some of those same techniques that we just heard about, image and video recognition. We're doing some very interesting work there, especially in neurodegenerative disease. And then also looking at kind of wearable devices. So many people are wearing them now. There's a lot of great data that comes off those devices, but there's also just a lot of data that comes off those devices. And so it's problematic to find signal and noise and make sure that you're understanding what the dynamics are that you're really looking for in some of those. And then when you look at kind of the data review and analysis phase of this, a lot of that is, again, kind of optimization and being able to do kind of appropriate signal detection, both for safety and efficacy purposes. So there's a lot of good opportunities in these spaces. So first one, kind of looking at that patient finding exercise. So on the right side is just actually some of the neural net maps and some of the other things that the AI model generates as it goes through patient finding. But what we do in this space is essentially offsetting the need for a lot of the chart review and a lot of the other things that go into patient finding for trials. Being able to work with different HCOs and being able to help them out in terms of finding patients. And really it is kind of about that right patient, right trial, right time. You can model out the way patient disease is going to progress. You can model out patient dynamics and being able to understand how they're going to progress through their healthcare system. That helps you with looking at not only when patients will become eligible for a study, but also looking at, especially we use this a lot in oncology and in chronic disease, looking at when treatment paradigms will have to change, looking at shifts from lines of therapy, being able to better kind of holistically handle the treatment of the patient and place trials in the right point in the patient care journey. So it's an important kind of dynamic. Many academic facilities have similar capabilities that they deploy themselves. We have capabilities internally that we use on internal data, and then also capabilities that we share with HCPs and HCOs that don't have access to those kinds of tools. They're very difficult to build, but they're very easy to implement. And so that's one of the things we try to make available with some of the facilities that we work with. So it's a really nice capability. It not only has it improved kind of the enrollment rate of studies, but it's also improved the ability for people. I mean, we've seen in some of the sites that we've worked with a 50-60% reduction in the amount of time that's required to review charts and improve the kind of experience of finding patients for enrollment in trials. So it's a really nice kind of capability. Then when we think about the other one that I wanted to touch on, which I'll spend a little more time on, is kind of the digital biomarker wearable devices, kind of that data collection in clinical research. So one of the biggest reasons to run clinical research is to be able to collect data and understand the performance of different molecules. And one of the challenges with that is some of the ways that we do it today. I mean, you can see in the example here with tolebrutinib, but in some of the ways we do that today, some of the measures that we use are somewhat imprecise. Or they have, if they're rating scales, they have significant rate of variability and other things that impact our ability to understand the underlying scientific accuracy of what's going on. And so we really try to take those kind of characteristics and see how can we deploy different types of AI models to improve the kind of precision and accuracy of what we're looking at. And some of those common areas in neuromuscular disease is things like gait impairment, movement disorder. You can start to see kind of important dynamics of the way disease progresses, and you can also start to kind of fine-tune the models to be very precise and to be able to find very minimal change over time. And so we see a lot of these applications being very much around, so it started out in wearable sensors, very much around kind of actigraph sensors and other movement sensors. Those are excellent, but at the same time, they're kind of limited in their scope. Movement sensors are they're subject to kind of the part of the body that you put them on. Actigraphs you can put on an arm or you put on a leg or around your waist, but they give you kind of limited data because of the dynamic of use. We've also started to do much more work with kind of image and especially video recognition. So you can imagine instead of just doing the 6-minute walk tests or other movement tests, you can take video of those activities and then deploy AI models to understand the discrete dynamics of the activities performed in the test. And that we found a lot of accuracy with those kind of capabilities, and there's a lot of work ongoing in terms of refining some of those, linking them to disease progression and endpoints to be able to better understand how they can kind of replace some of the representations, augment some of the representations that we have from kind of clinical records historically. So it's a really nice kind of dynamic, and it's improving the sensitivity of what we do. That's really kind of the goal with all of these things in the data collection space, is we really want to be able to generate as much data as possible so that we can most robustly understand the disease that we're trying to treat. So that is important kind of at the early stages to be able to understand natural history of some of these conditions. It's also important in the later stages to be able to understand kind of the progression of disease, the progression of treatment, and how some of those things work. So we've had a lot of kind of mixed models for these types of things. On a single trial, you see many different deployments of AI, which is a nice thing to see in this day and age. You'll see it kind of in that generative space, whether it's documentation or other capabilities. You'll see it in the patient finding space, which is very much about review, summarization, signal detection. You see it in the data collection space, very much in that same kind of image recognition, signal detection through medical records, being able to bring unstructured data into structured formats and being able to do signal detection in those contexts. and then being able to generate novel evidence through things like image recognition and pattern recognition through video. So it's kind of a lot of those different dynamics that Saverio went through, and kind of those different tiers of kind of modeling and AI. You'll see them kind of used harmoniously throughout different parts of the study. And my goal, obviously, with my team and with Sanofi as a company is to really make sure that we're deploying AI across the continuum of trials to be able to kind of maximize the operationalization of our trials and also maximize the evidence that we generate. So the goal is obviously to have as much information as we can as we move through these clinical cycles. And so some of this we can do early on with some of those digital twins and the synthetics, and then some of this we need to collect data in the trials and collect as much robust and precise data as we can to be able to move some of these things forward. And you're seeing some of that in kind of the publications. So just a simple example, you'll see a lot more publications from Sanofi in the AI space in the next 12 and 18 months, I can guarantee it. But this is an example of some of the work we did with the Actigraph and being able to look at kind of how can you take that wearable device, continuous stream data, so it's a huge data set, and being able to look at how can you understand different dynamics and signal detection of movement disorder from that device. Again, with the limitations of what Actigraph is, but being able to kind of mine through that and understand the dynamics of what's going on. And then one of the important follow-ups to this work was about 2 years ago, was that we were able to take those kind of models, and when you're looking at it from the patient's then medical record standpoint, you can link the Actigraph data with the medical record and being able to kind of understand tailored, personalized analytics on that specific person's disease progression, their health state, and some of the dynamics in their movement. So you can even see kind of differences between kind of baselines and other things as you look at these devices. It's a really nice dynamic, and as you start adding more kind of capabilities, more different modalities to these, you get a much deeper picture of what's going on, and it's a really nice kind of dynamic. It's helpful both in the preclinical space and being able to understand natural history, as I said, but it's super helpful in these clinical trials because we really want to be able to get that understanding. And we also really want to be able to work effectively with the HCPs that we're working with to make sure that in the phase 1 trial, we're deeply understanding the patient populations, so that by the time we're in phase 2 and 3, we've refined who those patients are, we've refined who the addressable population really is, and we're really progressing kind of that conversation around what these molecules can do. So it's an exciting space. It's a space I just came from a Sanofi conference about AI for 3 days and working with all of the physicians in the company to try to best understand how we can deploy these. And it's a space that I think is going to be not only growing, but is going to be really impactful as we look at the next 3 to 5 years. It's going to really change the way we do a lot of clinical research because we're seeing a lot better signal detection, a lot better and deeper understanding of what we're doing through these kind of executions. And so linking this with some of that molecule discovery on the front end is really kind of changing the game for what clinical research is, which I think is amazing because it's taking entirely too long and it's entirely too difficult to research molecules today. And so we've got to make this more robust and efficient as we get through it. So it's a great future for this kind of stuff. But as I said, a lot of capabilities that are coming and a lot of things that are going to be exciting over the next couple years. So, and I think I stayed to the 15 minutes. So,
very well, thank you. Before I ask if there is any question, I actually have a question for both you and Dr. D'Amico, because we talking of AI in clinical trials, many of us are dealing with rare disease, and we face the issue of placebo that we have discussed before. And I don't think regulators are ready to have trials without a placebo, but I think there is an opening in thinking that the placebo can be reduced and we can have a third arm of synthetic data and so on. And I wonder, do we already have an idea of what sort of numbers we need to validate this, because federated learning could be very useful in thinking on how many network centers we need to have proper data, to have reliable synthetic data to build an arm. And this is not only useful for reducing the placebo in the short term, which would already be a huge achievement, but many of our trials, in many of our trials, we only see results over 2 or 3 years, and it's unthinkable to have a placebo. So this could be a great idea to build, to have a design where we start introducing a synthetic arm in the first year with a placebo and then have a synthetic arm for a longer duration. So, well,
Yeah, I mean,
the question is for both.
In terms of, it sounds like the core question is in terms of like volume, like scale of the data that you need. So when we look at some of the things that we're trying to do with synthetics, especially in that area of placebo replacement, so we look at trying to be able to represent the population as a whole. So our goal is to get to about 0.1%, to train the models on about 0.1% of the addressable population. So if you're looking at that in certain indications, that would be a lot of people, as you can imagine. But if you're looking at it in some like rare disease or other specific indications, it can actually be a pretty approachable number in the tens of thousands or more. So it's the goal. The difficulty with some of those is, especially when you go kind of center by center, which is kind of how it has to work, you can get biases based on geography, based on socioeconomic status, based on other things in those areas. So we really try to get kind of cross-sectional views to train the models, which means you do need kind of like a percentage of the population. Is the way we kind of look at it.
Yeah, this is a very, very interesting point. So I would like to answer the two main points of your question. First of all, the amount of the data that we need to build a very strong and high fidelity synthetic data. So right now there isn't a threshold of data, at least from our experience, there is not a threshold of patients that we need for creating a high quality synthetic data, because it depends on the quality of the data, of course. So we reach, in the practical example, the same results in terms of fidelity for synthetic data in a clinical trial with a cohort. Of course, also me are working in the field of the rare diseases and also ultra-rare diseases. So we reached the same results in accuracy of synthetic data using, for example, 2,000 of patients and just 80 patients, because of course it depends on the amount of characteristics, the amount of endpoint that we are considering in the clinical trial, and of course the clinical question, because if the endpoint is a simple one or not so, let's say, without any different measures in time, maybe we need, let's say, a few patients. With few patients, I mean not 5 or 10 patients, but at least more than 50 patients. But of course, it depends again with respect of the complexity of the trial that we are going to replicate. So, in conclusion, it's not a matter of quantity, but it's a matter of quality of the data. First of all, and second of all, yeah, of course, we are approaching altogether in this new stuff of using AI and synthetic data in clinical trial. But I would like to stress the concept that synthetic data is not a substitute technology. So we are not going to substitute real patients or a real cohort of patients, but think about synthetic data as an integrative technology. Maybe to reach, for example, the amount, so how many clinical studies failed due to we are not reaching the enrollment of the patients. We reached the 80% of enrollment of patients instead of 100 of patients. So we can reach this gap in enrollment of patients using synthetic data, or we can explore some new clinical trial. For example, we can explore some inclusion criteria using synthetic data. Right now, there aren't any, let's say, study getting the approval in the European country, but there are several studies in which we are using synthetic data, of course, with the AMA that appreciate the use of synthetic data. And in particular, we are inside one very innovative trial in the field of neuroblastoma of children with Bambini Gesù in Rome with Professor Locatelli, and we are treated with CAR T cells. And in this particular and clinical scenario, the AMA, let's say, appreciate us in the use of synthetic data as a synthetic control arm instead of a single arm with historical data. So we are facing, in conclusion, we are now in the field of that regulatory bodies and regulatory approach asking to, let's say, clinical center to provide evidence and validation in which perimeters are synthetic data are really available.
Thank you. We actually still have 2 minutes before the next session. There is another question there.
Yeah, these are interesting presentations. I was fascinated by the presentation yesterday about placebos, and just to address the question that was just asked, I think one of the lessons from that talk is that maybe placebo is not natural history. And I've seen this over and over again in trials that we've done in neuromuscular disease. And so when we think about generating synthetic data, that's, I think, something that needs to be borne in mind. It'd be interesting to see what you all have to say about that.
Yeah, so you're absolutely right. And it's so interesting what you see in placebo data, because I mean, even placebo baselines and responses change geographically, and they change facility to facility. We do a lot of work looking at both kind of synthetic placebo groups, so taking cohorts of placebo patients from previous trials and creating synthetics, and then also, to your point, creating kind of what you'd call kind of like baseline population synthetics using medical records data from patients that were never part of clinical trials to be able to compare. And you do see significant differences between those placebo groups that were on trials and the groups that were just kind of natural history disease, general patient synthetic groups. And so there are kind of nuances to that space that have to be addressed, because you're absolutely right. They're not placebo patients, even in the kind of synthetics world, placebo patients are not equivalent to kind of broad non-clinical trial populations. They just have different assessments. So we do have to kind of figure some of that out, and we actually do have a program at Sanofi looking at how can we kind of explore what causes some of those differences and how to address some of that statistically in some of the analysis that we do, because it is problematic. You're right.
Thank you, and we wanted to thank all our speakers today for the lively discussions that grew out of this, and also clearly this is going to be the future, so I'm sure we'll all hear more about it to come.
Okay, so now for something different, something of an experiment which we hope you will engage in a positive spirit. So this session is going to be a debate, and I'm going to attempt to moderate the debate. Angela has kindly agreed to put forward the argument in favor of the question that we're going to define for you in a bit more detail. And John, being from Scandinavia, will argue strongly against the question. They've been instructed to take very polarized positions to help the debate. And I'm really keen, or we're really keen, that you do take part in the debate at the appropriate time. The organizing committee, it may have been one of the meetings I didn't attend when they nominated me to address this question, to treat or not to treat. When I saw it yesterday afternoon, I said, treat what? So they just said, well, it's up to you. So the question that we've tried to make I've tried to make it a little bit broader is, where a disease does have some form of modifying therapy, should it always be offered to all patients and families with that disease? And just to try to do a bit more context, so it's really positive. We are now clearly in an era of an increasing number of disease-modifying therapies for both genetic and acquired neuromuscular diseases. And this obviously creates really good opportunities, but definitely new challenges which our community are now starting to grapple with. It creates more complex decisions in clinical practice. Should such therapies always be given to all patients at all disease stages, whatever the risks might be? Some of the risks, as we heard yesterday in the genetic session, are not always fully understood, or their time course. Whatever the cost, whatever the size of the clinical benefit might be, often we're moving from diseases where there's zero treatment to a therapy where the benefit may be quite small. But is that okay? So maybe the answer is clearly yes, and there's no debate. Hopefully not. But obviously it's a bit more nuanced. I'm conscious that we haven't defined one particular disease, but despite that, we're going to have a go. And the way we're going to do this is based on that slightly less vague, but still fairly vague question. We want you to use the QR code and to vote now at the beginning of the debate what you're sort of, based on the experience you have before you came to this debate, what you feel the answer should be. Yes, we should always offer, always treat in all situations, or no. I appreciate this is a bit artificial. So I think, Gail, they should, or Donna, they should go to the QR code now, I think, everybody, and vote. We don't see the result, obviously, and then once that vote's completed, so I assume that'll be about now, we're going to ask John and Angela to put forward some considerations and some points. They've had no instructions, they're just doing this I think from yesterday evening. And they'll speak each, I think, for about 5 or 10 minutes. We then want to open it up, and we're really keen that people give examples, raise points, any that they think are relevant, and that'll be a sort of 15 minutes we have, I think, for that. And then we'll vote again and see if either John or Angela has changed your mind. So that's the broad plan, and I'm just going to sit there very quietly and listen to the debate. So over to the first of you. Thank you.
Thanks, Mike. Now that you've used sort of half the time, it'll be a short debate. No, but in talking to Angela the last couple of days, I think we agree on most things, but we'll try to disagree now. I'm sure all you guys, so before we talk about treatment, we just want to lay the foundation maybe a little bit about what are we going around doing? Do we all work smart? I know all you guys are smart, and most of the time you do the right things, but all of us don't do it all the time. I mean, are there things that we can do less of? And I'm already now starting on a negative slant here. You can see the world population is aging. You know, there will be more and more and more ill people in the coming years, and there will be fewer of us, at least in the Western world. And if we don't do anything, if we just treat, the health costs will just explode exponentially in coming years. So we should work smarter and try and cut away what we can. And here are some things that sort of some inputs on what we can do differently maybe. We should all have guidelines to follow our patients. Most of us don't have this for all diseases. If we have it, it's maybe per institution. And we can actually save a lot of trouble if we all had more or less the same guideline across really many sites. We could reduce a lot of redundant tests that our colleagues are making. We could collaborate much more easily. This is something we should really work on. And choose your tests carefully. Defensive medicine is all over the place. It's terrible how much money we're using on tests that shouldn't be done. Just last week, I was, I had some bills sent to me on my CITES antibody testing in my center, and it was just, it cost like $2,000, for an antibody test like this, and I don't remember the number, but it was a terrible number that we had used, and obviously you shouldn't use that much money on things like this. Think about it. A lot of you guys are doing EMGs, you get money for it. I mean, that is terrible in my point of view, that you get, that you actually link that you get money for doing a test, and if you don't do the test, then you lose money, or your hospital loses money. It shouldn't be like that, but that's how it is. And I know we are doing way too many neurophysiological examinations in this world. Stupid. How about thinking about how you see patients? Why should they come on a yearly rotation? I know a lot of you see them more variable, but do it even more on demand. Let the patient decide, to some extent, of course, because some of them want to come every day. But I mean, this could be done in a much more intelligent way. You can even also outsource some the way you are looking at your patients. Have your nurse look at the patient, the physio, or even your muscular dystrophy association help out to see them in between. And what about group consultations? Have you ever tried this? It really works great, and it saves a lot of time. There's a little bit with the booking, but apart from that, I did it first time 15 years ago because I was lazy. I had to convey the same message to some patients because they had been in a study, but it really opened up my eyes how fantastic it is because the patients love it. They talk to each other. You can address things to them in another way, and you save time. You save time in this way, and it's better. Of course, you can't do it every time you see patients, but every now and then do group consultations. And get rid of patients that are not neuromuscular. I mean, we all have these difficult patients, and we all do too little to finish them off. Honestly, they may be just 1 or 2% in your clinic, but they take up 20% of your time, right? We all have conferences and so on, but I mean, this is really something we can do better. Help your colleagues, to get rid of these patients. It's a huge resource that we waste on the wrong patients. And also be more restrictive when you get referrals. We shouldn't see all patients, Have maybe a check. So, like for instance, we have a checklist for small fiber neuropathy because it was just a tsunami for us, and now we have a checklist, and it has really helped in the referrals. And what about a patient that just has muscle pain and nothing else? Be more restrictive, and then. I don't know if any of you guys are doing it, we just heard about AI. Why not record and transcribe your medical records? This is actually possible now. That is really a great help. I'm sure we will all do it in some years down the line, but get started. It's possible already today. Is there anybody doing it now? Great. Fantastic. All right. The last two ones, I don't know how much time you save, because this is really saving time all the way through, but also making it better, right? But the last two, patient-reported outcomes and variables, using them in the follow-up is just that I think an improvement the video and phone consultations, saving time on part of the patient, maybe not so much for you. Just some inputs on how to do things smarter, quicker, save time, and I would really invite you to also come with some, if you have other suggestions. We should all work hard on this. So with that sort of introduction, before we go on to treat, I think I will hand over to Angela.
Thank you.
I'm clearly the shortest in this group. So I'm going to argue that we should treat. I'm going to argue the position that we should treat everyone who wants to be treated. And I'm going to argue it from two different sides of the coin. And then I'll give John a minute and a half to counter it. So I'm going to use the next half an hour, 45 minutes or so. Just kidding. So some of us in this room met, I figured it out, 35 years ago. And at that time, absolutely nothing we did basically was treatable. We had just taken IVIG from the immunodeficiency group. We started treating many different conditions. First MS tried it, and then neuromuscular. I don't think we did hardly any randomized controlled trials in IVIG, and yet I'm sure there's not a person in this room that hasn't prescribed it at least once, right? We just had discovered dystrophin. We weren't doing panel gene testing, and we were telling a lot of people that they had a really bad disease, and there was nothing we could do, so go home. Some of those diseases are very rapidly fatal, like ALS. Some of those are very long diseases in which they develop disabilities. I'm going to use FSH as on that side. And some of us were pediatric neurologists. I'm not. I'm an adult neurologist. And you had an even more difficult job. Your baby has SMA, and they will not live to see 2 years old. So that's where we're coming from, right? And we have spent the last 35 years doing the genetic testing, learning how to interpret even the very rare mutations, working with each other and with people in the pharma industry who are willing to tackle our diseases. So you've got to remember that we're not the favored disease group for pharma. We can't make them enough money, right? We are not, I have very bad MS envy because I think they got away with it, having MRIs discovered, so they got to get drugs approved much more quickly than the rest of us. But we're not the favored group of treatment. It's much better to treat a cancer. It's much easier to treat a cancer. We're a rare disease. We can't target a tumor and watch it shrink. We have to work much harder to do that. And we've succeeded. So we've succeeded in a few different areas. We haven't succeeded in everything. But if we have succeeded, as we have, let's use SMA, and now multiple therapies for SMA, or let's use a subpopulation of ALS. 2% of ALS is now considered treatable by disease-modifying therapies, tofersen. We've done it. We've started. We're going to get there in FSH. I am very confident that FSH disease-modifying therapies are just around the corner. We've done all this work in the form of clinical trials and clinical research. And the patients, the people with these diseases are so excited to get an opportunity to be treated. So who gets to decide whether or not we offer a treatment to someone carrying an SOD1 gene who is developing symptoms or has a fairly progressed disease? Are we on the side of those patients, and I'm obviously arguing we are, and that they deserve the chance to see a difference in their future. So disease modifying means that from the point of starting the therapy, you will change the course of the disease in a positive way. So I would argue that the fairest thing to do is offer it to all patients who have the condition, who have the disease. I'm working with genetics as the example. And I put my feet in the shoes of those people, regardless of their disability, having access to a drug. I consider that the ultimate in fairness. My colleagues would argue with me that we should stick with only those patients who were part of the inclusion criteria of the pivotal trial and saw a difference, statistically significant difference in that trial. And we all know this. Everyone involved in clinical trials knows that the inclusion criteria are set to see a benefit. They are narrowed to the patient population that is most likely to see a benefit in the course of the clinical trial. Usually somewhere between 12 months and 24 months, almost in all of our diseases, right? We will very rarely in our careers have another nusinersen event where the babies lived and the DSMB stopped the trial and the babies survived. That's a unique experience. But is that because we set those inclusion criteria for very pragmatic reasons, does that mean only those people who fit those very narrow inclusion criteria should get access to these potential disease-modifying therapy? I would argue that we should go broader once it's approved. The other thing that my colleagues argue with me is, well, that's not a big enough effect to offer. And my pushback as a treater is, well, who are you to judge how important that particular gain of function or delay of loss of function is to the person across the desk? So what do I mean by that? So I'm going to give you 3 examples, and you can decide which of these 3 you would have treated, all or none. So I'm going to give it, and all these people have been in the public eye, so I'm not revealing anything that's not public. So I have this lady who I still follow after 8 years, 7 or 8 years. Her name is Paula. She's done a lot of speaking about this. She carries the SOD1 gene, and she was included at my site. She's a fellow Canadian, but not from my city. She was included in my site in the phase 2. No, she was actually included in the phase 3 trial. She came to me with what you and I would consider very early ALS. She had mild weakness. She had noticed that she was unable to wear her high heels. Very big issue, by the way, all you men. This is like a big issue. You notice when you can't wear your heels. She was having to grab the railings when she got on climbed up the stairs. And she knew that like 100 people in her extended family, that's a bit of an exaggeration according to her, but many, many people in her large family tree had developed or died of ALS. And she had young kids. So she was admitted into the trial, so she met the inclusion criteria. And she's an easy one to make a decision on because she stuck it out. She continued to progress because, as you know from the tofersen story, the clinical benefit takes longer than the change in the biomarkers. And she was probably on placebo. And then about 6 months into the open label extension, she came to clinic and said, you know, I can walk up the stairs of the airplane now without holding on. And the next visit, which was a month later, I'm now wearing my shoes that I want to wear. The next visit, I'm back to teaching aerobics. And we were watching this evolve. Well, to me, that's an easy one. If that woman came to me now with tofersen approved and she had mild weakness, I would treat her. But the other two examples are a little more difficult, a little more challenging. Malcolm is the second guy who went in exactly the same time. By the time that he got in the trial, he was already in a wheelchair, a part of the time, for distances. He had still figured out how to golf. I don't know about golf, but there are people who will do almost anything to golf. And he was using, what was he doing? He was doing something with sea-doos. In Canada, we do sea-doos a lot on our lakes. Was still is able to use it, but he needed someone else on the equipment to run it. And he got into the trial just at the other end of the scale. He just barely got in. And this is now 7 years later, 6 or 7 years later, and he is still golfing. Before this, his trajectory was a normal one. He would have died in the next year and a half, 2 years, without ventilatory support. He's actually using the Sea-Doo by himself. He is still able to travel. And for all intents and purposes, he's completely plateaued. So he's on the drug. And for him, that plateau, despite the fact he uses his wheelchair for distance and he needs some help for various things, he still has maintained certain things in his life that are very important to him. So I think that is a reasonable person to treat if they show up, right? Here's the more difficult one. And this is the one that I would have you all think twice about your immediate thoughts. Here's a more difficult one, and there's a context. When nusinersen was approved in Canada, by Health Canada, even though the criteria were limited and the results were strongest in infantile, and we had additional data in juvenile, we really didn't have much data in the adult patient population. In Canada, it was a broad approval. It approved nusinersen for all people with genetically determined ALS. In Canada, the way it works is the provinces decide who actually get it. So you get a broad approval, and then rather than insurance companies making the decision, each individual province makes the decision. It actually comes down to 2 decisions, not 10, because there's Quebec and the rest of Canada. Most of you might know that. And so CADITH, this governing body, organizes for 9. And then INES, which is Quebec, organizes for Quebec. You know, big surprise. What happened was 9 provinces stuck to the inclusion criteria of the successful trials and gave access only to infantile and then eventually early childhood. 1 province, happens to be where I live, gave broad access. So then it came, what that means then is it fell on us as to whether or not we would prescribe it, right? So the upshot of that experience was that I was inundated by 20-somethings who were living in every other province, who wanted to go to university, who wanted to keep active, who wanted to try the drug, because I was the only place that they could stay in Canada and get access to the drug. So we had a huge number, in terms of what I normally saw, of these 20-somethings who were going to move to Quebec. They were going to leave their families, they were going to leave their systems because of the differential across the country. And we had deep arguments with it, and deep arguments. We had discussions over beer and wine between us, between colleagues in Quebec as to what to do, you know. I want to treat everyone who feels that they deserve the chance. Some other colleagues don't want to treat anyone unless they fit into the inclusion criteria. So we had the debate. So one of the young women who came to me, who could afford the drug, okay, because that's the last element I'm going to bring up, who could afford the drug. She said, you know, if this drug preserves my function in my right hand, I can go to university independently. Nothing wrong with my brain. If that hand function is preserved, I don't need someone to push me around. And I think that that is important enough for me, right? That do we treat that person so that that person can continue to be a functioning member of society and keep her independence? So I would argue I'd at least try it.
Okay.
I'd at least try it. Otherwise, I would argue that I am making a decision that is not mine to make. Is this quality of life issue important enough to that person to give it a try? So my argument is, with all of our therapies, I've just given you a few examples, but with all of our therapies, if we have enough evidence to prove the drug in a subpopulation, we should try. If we have evidence in a subpopulation, at what point do we go to a broader population? I don't know, but I think that's on us to do. And if this new drug is expensive, do we push it to who can afford it? Or do we help everyone get access? So my argument is treat, unless otherwise something indicates not. If you can access it, treat. Thank you.
Thank you.
All right. Sweet Angela, you should run for office one day. And that's why you speak long when you don't have data, I guess. And I guess these guys, they want data. And I'm really annoyed, you know, that companies conduct studies in children 4 to 6 years old, and then the drug is registered for everyone after that. And who says that, you know, when you are 72 and you had a disease for 72 years, that your muscles you're not building up anymore, and that you respond the same way to a drug? There's no data, and we have seen this in many cases. There's one there, Pompe disease. I have to give the credit, though, to Sanofi that they did eventually do an adult study. It was registered for adults, and I have to give them credit that for once that there was a study. It didn't work so well in the adults. And again, we can have a discussion, how long time should we treat adult patients with Pompe disease with ERT? A bigger problem is SMA, because there we have no studies in adult patients. There were just a few young adults in one of the trials, but basically we have absolutely no data. And so, yeah, what do we do? Then there's real world evidence, and we know that sucks. You know, that's that kind of data. Yet, in most countries now, we are treating nearly all patients. Then we make some criteria ourselves. And where do they come from? Just common sense, not science. We need studies before we treat patients. If we go on like this, because we can do it. We have very few diseases where we can treat with these extremely expensive treatments. If we go on like this in the next 10 years, we will absorb the whole healthcare system budget just for neuromuscular diseases. We can't go on. We need to do something. We need to decide on who we should treat and when. So And another example here that I put on is IVIG. We are over-treating patients with IVIG. That's an old drug even. That's not even one of the new ones. It's still rather expensive. MG should not be treated at all chronically with IVIG. There's no evidence for. Actually, there's evidence against it. Yet this still happens. CIDP patients, it's because we are not strong enough. The patients demand it. You know, you don't want to fight your patients. And we just tapered off our patients. It was a war zone, practically. But yet, half the patients in the end didn't need it. And this is something we know, there's evidence for this. But across the world, a lot of people just go on because it's easier and you don't have to fight anyone. There's a lot of treatments that we're doing. Stop treating when there's no evidence for it. So maybe I wouldn't take the extreme point that we shouldn't treat any patients at all, but and that we shouldn't treat patients with SMA that are older, because I'm doing it myself, I have to admit. But we're doing it maybe a little bit smarter, and that's not because of our center, but the healthcare system in my teeny tiny little country told the companies that there's no evidence, drop the price if you want to give this to the patients, because you have no evidence. So we are paying 20% of the normal price for treating our SMA patients now, 20%. And when we have treated in my clinic 35 patients, all the rest of the patients are for free. And we are treating 42 patients now. So the last 7 patients are treated for free. And we can go on if we can find more patients. Probably people would start sending us patients, I think. We should think about, and the trouble is that we are in different countries and we don't have one voice. But we should all try and think in this way, that if we want to break evidence and treat patients anyway, at least we should talk about the price. All right, I think I will hold here.
Okay, so thank you to both our speakers. So Angela clearly is the good cop. She really cares about her patients and will be running for office. And John, on the other hand, is maybe the bad cop, may even want to terminate non-neuromuscular disease patients, but actually made good points about the need for hard evidence, that there's overinterpretation of inclusion beyond the trial, inclusion, and the opportunity to negotiate, to get prices down where the indication is beyond the trial entry criteria seems like a really good point to me. So we have some time to open it up. I'm keen that people make comments. Also, I should mention the point that John made about working smarter, which obviously I think is highly relevant to all of our clinical practices. So any points from Tony? Good. Would be disappointing if Tony didn't have a point.
Angela. So just because a drug is approved, it needs to be offered to everybody. What about drug X? Say drug X in ALS. That was studied in an ALS, found to be negative. Subgroup analysis of a very, very fast progressors showed that it was approved. Showed maybe there was a 2-question benefit. And then somebody says, oh, that's a 30% functional loss, which I think you're smart enough to know it's not. Doesn't mean motor neurons, 30% less motor neurons died because they had 2 questions. But now it's offered to everybody in the population. So you said there wasn't a study that showed, but there was a study that showed in all comers it didn't work. So you would offer that knowing the large study to people that aren't very fast progressors. Now there's a second study that was done and then showed it didn't work again. So are you saying just because one study, when other studies failed, and very fast progressors should be offered to every single person? And can you tell me that you think in the patients who you see that it's really made a significant difference? And with the cost that it should be offered to everybody.
Okay.
You can become yourself now.
No, do I stay in character?
Stay in character. Stay in character.
Let me stay in character. Not that anyone in the room knows what you're referring to. So here are the arguments in character. There has been 2 or 3 surveys, one of the clinicians and one of the patients, and there will probably be some work done on this, and that is what is significant to a patient, as opposed to how much benefit does minimal important difference. What is the difference between what you could objectively record as a minimal important difference and what is significant to a patient and what is significant to an expert panel? And in fact, in this functional rating scale, those 3 groups come up with different numbers, right? So the patient groups say 1 point difference in the ALSFRS score, if it is a relevant muscle, a group like gross motor function, is important to them. Same as a young adult with SMA that just wants her hand to preserve her hand function. The clinical experts say 2.4 difference in scores on the ALSFRS is relevant and enough and that's where a lot of our stats are coming from. And then a methodologist has just proposed that you need to have a 3.8 difference in the ALSFRS score to be clinically important. So I obviously would like to treat those who want to be treated, at least short term, in this scenario. So the patients say 1 point difference. And in the subgroups, you can work with or without the subgroups, but 1 point difference. So they would argue, until you look at the second study, that as long as we have 1 point difference, that's enough for me to take the drug. So that's the position that the treaters take.
Okay. There's a question there towards the back on the left, and then the one here at the front. Great.
So this is sort of a real-world question for you guys to stay in your themes. I will say, before I ask the question, the debate reminds me of when I was in 8th grade. I had to participate in our junior high mock government. And I had to represent a political party, and we had to draw lots, and I got the Conservative Party. And I had to be against every bit of spending known to mankind, and I ended up getting like 0 out of 600 votes. So I have a little PTSD, so I have some empathy here. But the real world question is, and this really happened, it's a real story, but I wrote a couple of papers with Peter Anderson like 20 years ago, and we've stayed close over the years, and he's an ALS geneticist doctor in Sweden. And we opened up a genetic SOD trial in San Francisco, and Peter called me up and said, I got a bunch of patients from Sweden who'd like to come live in San Francisco. And they moved all the way across the world, and we put a bunch of patients from Sweden into our San Francisco trial so they could get these highly complex viral vector anti-SOD1 injections. And it was sort of heartbreaking because these guys are trying so hard to fly across the world and get this treatment, and it's a safety study, so they're getting crappy little doses at the beginning. And on the other side of the story is we have tofersen in our disease, and those of us who've practiced a lot of ALS, which includes Angela, even though the clinical data from that study is pretty crappy, and we're basing our belief on neurofilament, we have seen some cases that truly at the bedside get you to believe the drug's working for the reason Angela told us, but also because you have these patients with these horrible mutations, and even though you don't have a control, in the past those patients seem like they would have died quickly. And as long as you're giving tofersen, they do well. But in Sweden, you cannot get tofersen. So every single one of these patients was lamenting appointment after appointment after appointment that they can't get tofersen in Sweden. So I'm interested in your roles, and not only your theoretical roles here, but your actual roles of who you are. How do you square these guys not being able to get tofersen in Sweden, and how can they get tofersen in Sweden? And how do you square these guys flying all the way to San Francisco to get their heart broken? So that's sort of the question.
John, you first.
Well, it was an ALS question, wasn't it? I think that, it happens all the time, right? With all these trials that we are running, it's so difficult to get the treatment, and now they travel because it's reimbursed. But I mean, also when you do run clinical trials, we just done a gene therapy trial. We have 200 patients lined up for 6 slots. That's how it is. And how can you fight what local governments decide on whether you reimburse or not? It's kind of out of our hands, isn't it?
What do you say to your government in Sweden?
Well, I mean, the thing is that mostly they're not really affected by what you say. They have a body, and the bodies have different names in different countries that are more or less closed and are not really affected by, the community, patient organizations, or even us as a full body. That's my experience.
Okay, we have time for one final question, then I'll give you the vote.
It's just a quick comment. I like this version of my friend John as an opponent. He actually reminds me of politicians we have in office at the moment, which I better don't mention here. Anyway, I like the way you tried to kill in 3 minutes 20 years of building up care recommendations. If we were waiting for our patients to come to clinic when they wish or when they feel they need, not performing annual cardiac or respiratory tests, we would probably kill half of them and kill 20 years of building up care recommendation and prophylaxis and so on. So well done, John.
Okay, so thank you. So we're going to just go to give you the results of the vote. So before the debate, 67% of you felt that we should offer treatment to every patient, and 33% felt no, we should not. But John has had a significant effect because he's increased the number of no's from 33 to 37%. So I want to thank both for great presentations, and thank you all. So we're going to break now for coffee. We'll regroup maybe 10:35. Thank you. Well done. Okay.
So for our first speaker is going to be Dr. Chad Heatwall. He's a professor of neurology and director of the Center for Health and Technology at the University of Rochester Medical Center, and he'll be presenting on the lessons learned in terms of regulatory success from clinical trials to drug approval.
Thank you so much for that introduction. It's just a real pleasure to be back here in Stresa. And thank you for the organizers for really putting together such a successful and enjoyable meeting. So I'll be taking a couple minutes here to talk about my experience with clinical trials and how they've led to drug approvals. And before I start, I'd just like to thank our sponsors over the years. We've really been blessed and lucky to work with a lot of different organizations, from industry to foundations to patients and government entities. And it really takes a village to do good, successful clinical trials, and you can't do it without many partners. And so I'm going to build a little bit on what was said yesterday, and just amazing talks about clinical trial units from Valeria and others. And you get a sense that it is very, very difficult to do good clinical science, to do good clinical research, and more so, it's difficult to really get to regulatory approval. And developing, validating, approving, and disseminating worthy therapeutics for humans is challenging. But we don't get into this because it's easy, we get into it because it's worthwhile. And this slide gives a little bit of an idea of how hard it is. And so it's estimated that it takes approximately 5,000 to 10,000 compounds for every one regulatory approval. And it takes a long time. There's multiple phases of research, and there's a lot of opportunities for a trial or a therapeutic program to fail. And if you look at the success rate, so in 2024, there were only 50 FDA approvals and only 46 EMA positive opinions. And if you look at other areas of the world, that's a pretty rare number. So if you look at FIFA World Cup hat tricks, there's only been 54 of those in the history of the world. So, again, it's a little less frequent than that. The naked eye supernovas in the history of mankind has only happened about 50 times. The 50 home run season in U.S. baseball has only happened 51 times. And there's only 50 discovery of new mammals every year. And so this is as rare as hen's teeth, and it really doesn't happen a lot. And it's complicated. It takes a lot of people. These are estimates from this OncoDaily website, but phase 1, you have 20 to 100 people. Phase 2, you're dealing with 300 people. Phase 3, up to 1,000 people. And phase 4, many more. I think the numbers that are provided at the bottom are actually pretty conservative. 4.5 million for phase 1, phase 2, 11 million, phase 3, 22 million. I think depending on the study design, those numbers can be actually quite a bit higher. And so what you see here is the list of FDA approvals by year, and it fluctuates. There's a lot of things that go into that. There's political factors. There's certainly other factors as far as the dynamic for developing what the FDA is doing. And really, most recently, we've been doing okay. You look in prior years, and it's been as low as 19 or 18 drug approvals. So how do we do in neurology? And I guess we do okay. And so here's a graph. Far and away, the area of medicine that is most productive in new drug approvals is oncology. And you can see both the 5-year and the 1-year total here. But neurology actually does pretty good. This is the forefront of technology in a lot of instances, and it's the last frontier. I've heard that neurology been described as that. And so on average, neurology gets about 10 to 11 drug approvals per year. Again, the last year was not as productive, about 4 approvals. So the NIH decided that they would provide a very easy to understand and useful diagram on, how do you actually get a regulatory approval? And so they published this. You can take a look at it. I've looked through it pretty thoroughly. It is complicated, and they break it down into 6 different units. It's target ID, biomarker, outcome development, narrowing down lead identification, lead optimization, Then clinical research is number 5, and then the regulatory process is number 6. And these are all very difficult and very challenging, and it's needed to have deep expertise to navigate this. And so the NIH then said, well, we'll make this even simpler. Here you go. And so this I found very entertaining. It is called the Interactive Translational Science Map. And it's a list of about 1,000 bubbles, and you can click on it, and it will tell you how they interact. And there's lines to all the other 1,000, and it was almost comical how complicated this was. And I noticed that in April 2022, they actually dropped this science map, so you can no longer view this. But I got a screenshot of it before they dropped it. If you boil it down, this is what it comes down to. And again, it is multiple partnerships across multiple venues to make therapeutic developments happen. And so this really starts with the patients and ends with the patients, but also there's involvements of academic groups, foundations, the FDA, NIH, philanthropy, clinical research organizations, the government, payers, industry, and it's sometimes CHET. And so I'll talk about CHET in a little bit. So it's not just important to have a good therapy in hand and a good study population. You have to have appropriate and well-validated therapeutic assessments. And there's many different types. The gold standard, of course, is survival. But in our chronic neurologic diseases, that is not always a very viable outcome measure. They survive for a long time, and it's not possible to run clinical trials without using a lot of finances. So the other assessments often include biomarkers. They can be easy to use. They can change early. However, they have unknown relevance. There's functional state. Again, we love this in neuromuscular diseases. How strong is someone? How far can they walk? They may lack responsiveness to change. We all know 6-minute walk distance is a great and well understood outcome, but it's not always responsive to therapeutic intervention. And then lastly, we have patient-reported outcome measures, and these can provide really how is this meaningful to a patient, which as a clinician is quite important, but they have to be developed and validated in a systematic way that is appropriate. And so the FDA has different things to say at different times by different people. But in general, they define that a clinical benefit is a favorable effect on a meaningful aspect of how a patient feels and functions. They say that clinical benefit may be measured as an improvement or delay in the progression of disease. And so that is what is so challenging in neuromuscular disease, is that a lot of times these are chronic, slowly progressive diseases. And so to really statistically detect a progression of disease can be challenging. Indirect assessments need justification for its value as a replacement of how a patient survives, feels, or functions. An observed clinical benefit is described in a labeling as a claim using words that represent the concept measured. And this should be easily meaningful and understandable to both prescribers and patients. And sometimes that can be a challenge, and it's not always well specified by the FDA. So to say a couple words about CHET, this is where I work. It's at the University of Rochester. It's called the Center for Health and Technology. It's about a 100-person entity, and our mission, quite frankly and simply, is to advance human therapeutics, health, and knowledge through exceptional people, skillful research, and partnerships. And so we've conducted over 155 multi-site therapeutic trials over the last 30 years. These trials have involved over 45,000 participants and has resulted in 12 FDA drug approvals, actually 11 drug approvals and 1 device approval. And the most historic and longstanding unit of that is our Clinical Trials Coordination Center, which is really, it was set up by Irish Olson about over 30 years ago to really study movement disorders and therapeutic studies in movement disorders. But we have other entities that, again, are essential for conducting clinical trials. We have a clinical materials service unit that ships drug in a regulatory fashion. We have a CHET analytics group. It's great to hear the AI talks today. The Chet Analytics does deep dive statistical modeling and predictive modeling in neurologic disease. We have an innovation group that focuses on remote trials and novel devices. We have contracts with Apple and Apple Watches. Chet Outcomes, which I started about 20 years ago, and Chet Health, which is getting our innovations into the community to help everybody. Like I said, 155 trials. We're currently running about 40 trials. Done this in 45,000 participants, 20 scientific publications a year, if not more, and over 30 years of operation. And our FDA drug approvals have been mostly in movement disease. 11 of the 12 have. 11 of the 12 were drugs. 1 was device. But we have had experience in running successful regulatory approved trials in neuromuscular disease, specifically in a drug for periodic paralysis in 2017. And this circle may look like the circle that I showed you earlier, which kind of sums up what is really needed to successfully run a regulatory study. And again, we are an academic CRO or academic entity, but we're very externally focused to industry, foundation, patients, government, FDA, NIH, and other academic groups. We mostly run industry studies, but if it's a multi-site trial, we also assist academic groups with their work. And the most longstanding and really the powerhouse of Chet is the CTCC, or the Clinical Trials Coordination Center. And this is soup to nuts, helping academic groups and industry run their studies. It involves study startup, monitoring, data management, clinical trial rescue and recovery. So we've rescued studies from large for-profit CROs. And really that goes to play that we have the deep expertise in academic groups and knowledge in individual diseases, and we know partners like yourself who recruit well, and so that is a tremendous advantage in running studies. And then statistical analysis. And it's really difficult to do this in a regulatory fashion. And so we have 2 full-time quality assurance people that run consistent and regular audits to make sure that we're satisfy all the FDA's needs. We have over 100 SOPs that were reviewed on a regular basis to really make sure that everything is compliant. And so that degree to get to that regulatory approval is quite challenging. And we also have the CMSU, Clinical Materials Service Unit, which ships drugs, so things like the Healy study, the ALS study, we ship all the drug for that, as well as many, in fact, 90 drug and device multi-center clinical trials. And it sounds simple, but it's not easy, and it has to be done correctly. And so these little steps and little things are critically important and can be quite challenging to do correctly in a regulatory fashion. And then one of our units is Chet Outcomes. And so this was started by myself about 20 years ago. And really the motivation is to give the patient a quantifiable voice during clinical trials and industry trials. And so we set out to really make the best instrument possible that can really detect and measure over time a patient's perception of their own disease burden in multiple different areas. And again, this was designed for industry trials, but over the years, these have been utilized in academic studies, government studies, and foundation-sponsored studies. And so why do we need these? So a good outcome is critical. If a treatment is curative, it doesn't matter what outcome you do. If a treatment doesn't work at all, it doesn't matter what outcome you use. It's not going to help it. But if it's in that middle zone where it provides some therapeutic benefit that's perceivable by the patient, it's really important to have a good PRO to do that. And so why disease-specific? Well, they provide a unique window into a patient's perception that you may not be able to get in other ways and through other functional measures. The second point is they are thought to be more responsive. As a clinician, we know that when we're in the clinic, a patient will say, hey, I feel a little bit better in this way, or I'm a little worse in this way or this way. And sometimes we cannot observe that through other signs or examination features. And so it gives us a window and a way to measure treatment early. And PROs have been used by the FDA and EMA for a long, long time. And so before 2009, they were actually involved in about 20%, 21% of all FDA drug approvals had a PRO in the label, and then 23% of the EMA. And about that time, the FDA said, okay, it's kind of the Wild West. There's a lot of PROs. Many of them are bad. Many of them are not responsive or meaningful to patients. So they put an industry guideline out, or a guideline for industry on how a PRO should be developed. This has since been slightly modified, but has not changed so much from the original 2009 guideline. And they set the bar extremely high. So there's many things, and you see here 9 different components of what a PRO should do. It has to be meaningful, reliable, responsive, low patient burden, no redundancy, have a high level of validity. And when the FDA says validity, what they really mean is 7 different types of validity. They require studies to demonstrate all of these in their PROs. And if I had known how much work this was going to be to do this the way they want it done, I probably would have run away, but I'm glad I didn't. And so the CHED Outcomes Division is really set out to develop and validate highly sensitive, clinical meaningful outcome measures to detect therapeutic gain during clinical trials. And the first time we did this, it took about 5 years. We're now down to about a year per instrument, soup to nuts. And we have a large group to do this that involves psychometric researchers, biostatisticians, outcomes researchers, patients, and expert groups. And I won't go through all of the steps, but it's very extensive, it's onerous, it's complicated. And it involves large-scale patient input. So we put large-scale patient input in the front end to make sure that the items and the subscales that we're using are relevant to those specific diseases. And so that involves qualitative interviews, it involves cross-sectional studies, a lot of psychometric analysis. And so why? So why disease-specific? There's a lot of outcome measures out there that are generic. There's PROMIS, there's NeuroQual, there's SF36. Well, there's a higher reliance and relevance and content validity when you do disease-specific. We know that our neuromuscular populations have specific symptomatic areas that are unique to their particular population and wouldn't be the same as lung cancers or Crohn's disease. More importantly, when you make a disease-specific outcome measure, you can reduce sample size, you can have a more responsive instrument, and patients actually comply with it in a more easy way when they are not having to answer questions that are irrelevant to their population. And so this is a partial list. We've developed instruments now for over 30 diseases, and it really started in this group. And so most of these started with neuromuscular disease, and you guys have probably heard about the MD high and the SMA high and the ALS high and the FSHD high and the CMT high. And so we've kind of expanded off of that list, 30 different diseases. We do have a website if you want to know about individual ones, but we've expanded beyond that, movement disorders and also other diseases. And they're used for multi-center and multinational trials, so we have over 38 different languages that they've been translated into. And some languages are not only specific to that language, but the region of where they're spoke. And we've done many of these projects with Filaria for our Italian translations for many of our instruments. And so the other key aspect about these, and this is important in regulatory approval, is that they're multifactorial instruments. And so they're composed of multiple subscales that measure a very granular symptomatic domain for each of those diseases, and it's depending on the disease. And that's important because the FDA is not quite so interested in a global improvement of health through HRQOL, but they want to know, is there an improvement in a patient's perception of their myotonia or of their own mobility or their upper extremity function? And so all of our instruments are scored from 0 to 100 for the subscales and then have a composite score as well. And that's important because if you're not going for a genetic treatment or a genetic cure, let's say you have Tylenol and you're looking for another use for your drug, and you want to test a myotonic dystrophy, there's a pain-specific subscale for that. So for instance, in our ALS measure, every time a patient completes that, they get a score from 0 to 100 in each one of these issues. And each one of those subscales was identified by over 1,000 patients working with the CDC who said, these are the issues, the 15 issues that are most important in my life as they're related to ALS. And so if you have a therapy that's looking to change fatigue in this disease or pain or sleep, you can use the subscales. Our first instrument is myotonic dystrophy health index. There are 11 publications on this particular instrument, and it's provide a useful guide for industry and regulatory approval. There were several posters that were in the session yesterday, actually, that utilized this particular instrument. And it's been identified by the NIH as really the only instrument that's highly recommended to measure disease burden over time. And the interesting thing that the NIH said was that they are recommending it as a primary or secondary outcome measure. And so that's important because in the past there's been reluctance to use a PRO for a primary outcome measure. And we think that it's not unreasonable to do so. The patient's perspective of their disease is paramount. And so how do these correlate? So ideally, a patient's perspective of their disease should correlate with clinical function. And so we've done studies to really look and prove this. And in a clinical study, you obviously want a functional measure, you want a patient-reported outcome measure, you want a good biomarker. And in an ideal world, they correlate and they all together support a therapy. And so we've shown that the MDI, for instance, we have data in other diseases as well, correlates well with breathing, impairment function, strength, and ambulatory state. We've also done head-to-head with generic instruments, the INQUAL, SF36, SF6D, and what we found is that there's a stronger correlation with strength and ambulation and impairment, muscle impairment scales with the disease-specific instruments compared to the generic and semi-generic instruments. We've also shown that our instruments have a stronger association with disability compared to these instruments. And we've also shown that patients prefer our instruments, even though they're slightly longer. And again, it's not the length of the instrument, it's the quality and the content of the instrument. And so when you ask a patient, hey, how's your marathon time, and they're in a wheelchair, it's an extremely abrasive question. And so when you ask questions that are relevant to a population and appropriate, it's easier to complete. And the other thing I'll say is the layout is important of an instrument. And so because of the simplicity of our instrument, you can complete the questions faster than other instruments. So on average, patients complete 8.3 questions per minute of our health indexes compared to other instruments that are much fewer questions because they're not having to change questions, change Likert responses, which can add burden to the patient. And we've done sample size efficiency calculations, so we know the MCID of our instruments. And so if you design a study using the MCID of our MD High, you would only need a study that had 36 patients. And this is in contrast to if you use something like the 6-minute walk distance and its MCID, you would need 109 patients. So it brings efficiency to trials. And they're responsive. So this is a data from our Parkinson's longitudinal study that we're doing remotely through Chet. And so what we noticed is that our instrument can detect disease progression over a short time as six months, and this is over generic instruments in this field. And so what I was saying before with the FDA saying you need to detect change in progression of disease, this gives you a shot to actually do that. And then we've developed mechanisms, longitudinal anchor-based methodology, determine the minimal clinical important difference or minimal clinical change. This is changing from the FDA's perspective of not how to do it, but how to display the data. And so we're modifying our techniques to satisfy that as well. And so we have those values. And there's been 5 pivotal studies or important studies over the last 2 years that really demonstrated the ability of our instruments to detect therapeutic gain in a clinical trial over generic instruments. And 2 of these, Dyne and Applied Therapeutics, you can look at their press releases. They also had wonderful posters yesterday demonstrating the CMT and MD High's ability to detect therapeutic gain. The Starfish study, which was a single-site study that we presented here at this meeting last year, and the last time is at Stress as well, also demonstrated therapeutic benefit to our therapeutic mechanism, and the FSHD high was superior over the PROMIS. And then Spinraza, in response to that awesome debate right before there, and Craig Zyman, is Craig here still? He had a nice presentation yesterday. Also showed that the SMA high was able to detect therapeutic gain in response to Spinraza in adults with that disease. And then lastly, Neurocrine. So Neurocrine received a FDA approval for valbenazine in Huntington's disease in 2023, and that was through the CONNECT study. And that was run by Chet as well, and it was really to evaluate this drug to reduce chorea and HD over a 12-week period. And Chet ran this study in the midst of a COVID pandemic, and I think that really is a testament of our personnel with deep experience. They were able to kind of navigate those challenges and successfully complete that study, which was later published in Lancet and resulted in the FDA approval. But the interesting thing about that study is their primary outcome measure was successful, and it was a measure of chorea. Their secondary outcomes, which were PROs, did not perform as well, but they happened to put the HD high in there, which has subscales that basically measure abnormal movements, which is how that drug works, and that was able to detect therapeutic gain over placebo, and ultimately they received an FDA label for that. And then I won't say too much about this. I think you should nab Craig Zyman about this as well. But again, there is a study, and this was presented at the AAN, and hopefully the paper is forthcoming of the full study. But basically looking at, how does Spinraza affect the lives of patients with SMA who are adults? And so the SMA high was able to detect a therapeutic gain there. Also, the revised upper limb module at 6 months, whereas the Hammersmith did not do as well. So what are the conclusions? I think just 3 points here. The path to regulatory success is challenging, long, and quite complex. Partnerships with organizations skilled with rare neurologic disease increases the probability of success. And lastly, it's really important to select the right outcome measures. Proper therapeutics, patient population study design, and the outcome measures together are really the ingredients for a successful trial. So I'd just like to thank you for listening, and thank my group back home, and many of which are here today from Chet. Thanks.
All right, and we have time for 1, maybe 2 quick questions. Okay, maybe I'd missed it, but how did you calculate or measure the minimally clinically important difference or the change?
Yeah, so it's a great question. There's a lot of different ways to do that, and it's unclear at this point the exact mechanism that the FDA requires. However, they've often and consistently said that statistical methodologies using standard deviations are not appropriate ways to get the MCID. And so the way that we've done it in the past is we've used anchor-based methodology in a longitudinal study to compare to changes in our instruments. The other way to do that is actually look at changes in functional measures over the same period of time and how those correlate with changes in our subscales, for instance.
Wonderful. All right. Thank you, Dr. Heatwall.
Thank you, guys.
And our next speaker is Dr. Eugenio Mercuri, and he is a professor of pediatric neurology at the Catholic University in Rome, where he specializes in neonatal neurology and neuromuscular disorders. And he'll be talking to us about the lessons learned in Duchenne muscular dystrophy.
Thank you. Well, and thank the organizer for giving me the opportunity to discuss this, which I think it fits very well with the rest of the program. These are my disclosure, and we go straight into the topic. We have just heard how difficult it is to get a drug approved, especially for Duchenne. This has been particularly challenging in Europe more than in the States. And several trials in Duchenne failed to meet the primary endpoint, or at least to reach statistical significance, even when, as clinicians, we can see clear signs of possible efficacy. And it has been nice to see how we often blame the outcome measures for the failure. These are the results, just an example of the EMBARK Part 1, or the one-year results. You can see that the primary outcome measures, which was the North Star, failed. It was trending in the right direction together with all the others, but didn't reach the statistical significance. And there was also a bit of concern or discussion, let's say, on why while other measures were reaching statistical significance, the North Star did not. So why don't we see concordance between measures and so on? So at the end of the review of this data, the North Star was blamed for it. I could say it in a different way, what people felt about the North Star when they saw these results, but the reality is that they felt that the North Star was not appropriate as an outcome measure. And then the 2 years results came out, and the North Star was very significantly statistically significant. So the North Star went from it's useless to the new flavor of the month. And there was a bit more concordance, but still not full concordance. So there are these 2 points which we have to keep in mind. So what did we learn from the EMBARK was just an example. I could have brought you other examples which were very similar, and there were a number of mistakes that I think we understand much better now. The first thing is that I have been involved in several advisory boards, and very often people ask me, do you agree that we use the North Star or the time to rise or whatever as the primary outcome measures? And they ask me if I agree before they mention who they are testing, the duration of the study, and so on, the population study, the age, and so on. So very often outcome measures have been selected in clinical trials before defining the population, just because they were working in another study. And then we had the other part of it, that you spend a lot of time in an advisory board and you decide what is the right measure for the right population, for the right duration of the study, and so on. And then eventually, when you see the protocol of the study, you see that they have also included younger, older, weaker, stronger patients, just because the commercial team said, for the label is much better if you also include the 3 years old or the 12 years old, and so on. So all the work you do in building a trial design which may work is completely lost. And so I think we have learned now that there is really no functional outcome measures that we believe as relevant across the entire spectrum in Duchenne, from the very young to the older boys. And we have learned that there are a number of elements that we have to take into account. Age is one of them. These are studies that we have been publishing for the last 15 years, nearly 20 years, I think now, where it's obvious that in Duchenne you see an initial phase or stability or even improvement, and after the age of 7 you see a decline. So we do know that if you perform studies in very young children before the age of 7 or in older children, the trajectories are completely different. And while at the age of 7, we show, the second point is concordance across measures. While in the children older than 7, if we use timed items and the North Star, you can see they all trend in the same direction. A decline in the North Star is a reduction in score. In the timed items, increase in times is a decline. So even if the lines go in a different direction, all three are trending
with the same meaning. You know, there is a decline after the age of 7 on all the measures we see. Before the age of 7, this is trickier because there is a lot of variability and the measures are not concordant. So I will show you now some data, and I want to thank Giorgia Coratti and all my team because we have been working hard in bringing this data together. I'll show you some unpublished data today. And we have been able to see with a very large cohort, which is the results of an international collaboration, the lack of concordance in the children below the age of 7. These are children at the age of 4. What I will show you now is not only that there is no concordance, but also for each individual year of age, things can be different. These are children who were seen between the age of 4 and 5 and who were followed for a year. And you can see that at age 4, there is some concordance because these children at this age are still improving on the North Star and on the time to rise from the floor and on the 10 meter. So the very young ones show improvements on everything. If we look at children who are just slightly older, at age 5, we can see the children are still improving on the North Star. They are a bit more stable on the 10 meter, but there is already some decline on time to rise from the floor. So time to rise from the floor is the first measure when we start seeing decline, even at the time when the other measures are stable or even improving. At 6, we have that North Star doesn't improve anymore, is stable as is the 10-meter walk test, and there is more decline on the time to rise from the floor. So if we are using different measures and we are selecting population with different age groups, including 4 or 5 or 6, can make a difference. And depending on the mechanism of action and on the hypothesis, you may select a population which is in decline or not, and therefore choose one measure over the other. And this is a graphical way of showing that at 4 there is concordance. This is measured in terms of velocity, so also the timed items are on the right. And you can see how quickly the time to rise becomes discordant with the other 2 measures. After the age of 6, this discordance is less obvious. So the one question was, why is the North Star following a different pattern? Why do we see an improvement on the North Star even when the other scales already show a decline? And the answer is that the North Star is a functional scale which includes a lot of functional aspects which are also related to growth and development. These are very young boys who are growing. They are learning how to do things. And a few years ago, we published a study when we looked at what is the possibility that untreated children, this is natural history, may show an improvement between 3 and 6 years. And you can see from these numbers here that the improvements can be on average 7.3 at 3 years and so on. They decrease with age, but they can be as high as 13 points, 16 points, 14 points, and so on. So this is scary if you are designing a trial with a placebo, thinking that your placebo may improve that much. And we have looked at what are the possible factors which may affect this increase in scores, and the site of mutation and the brain dystrophin isomers are very important. I'm sure you all know there are different brain dystrophin isoforms which are associated with different sites of mutation. Patients with mutations after exon 44 and or 63 have additional multiple involvement of multiple brain dystrophin isoforms with increasing severity of cognitive aspects. And this is a very elegant paper published by Francesco Montoni's group a couple of years ago, showing how the different subgroups, subdivided according to brain dystrophin isoform involvement, reach different peaks at different ages, and they have different lines of decline. And this is what we saw a few years ago. If you look at this graph, you see the changes on the North Star in children who have a different baseline on the North Star. And it's obvious that there is a trend which is very obvious, but also if you look at this data, there is there is a cluster of patients who show a lot of improvement. Who are these patients? This is counterintuitive. These are the patients who have a lower baseline. So the children who start with a very low baseline are not weak, are just children who have a delay. And because this is just a delay, they will eventually acquire the skills, and these are the ones who can show improvements over 6, 8, 10 points, and you can see. So this is different from what we will see in older patients. So baseline is important, but is with a negative linear correlation, because the one with lower scores will have better outcomes. So there are a number of issues, and in the younger children, we should not forget that this is the time when we start steroids, and steroids will cause an increase in the scores, especially in the first 6-12 months. These are our results showing that the difference between children who were introduced to steroids or who were already on steroids were very important. And when we look at all this together, looking at the possible effect of steroids, age, and baseline, we saw they were all significant, and steroids was had the highest degree of impact on the trajectories. Even after the age of 7, there is some variability. These are the individual lines of what we see in after the age of 7. They are all declining, but they are not declining with the same speed. And over the years, we have tried to find whether there are other elements which may help us to identify the children who are decreasing more rapidly than others. And time to rise from the floor is an important factor. If we subdivide the children according to their time they rise from the floor, below 5, 6, 7 seconds, we can see that this has a large effect, and we can identify different subgroups. I'm not going into details. And I think it's very important to try to understand that if we want to use an outcome measure for a trial, we should not just look at the outcome measures, but also at a number of other elements. And this is what happens in Duchenne. This is the 6-minute walk test, but we could have this with any outcome measures. And we know there is a time, even after the age of 7, when children, but especially below the age of 7, when children may be more stable. Then there is a time when there is some decline, and theoretically, if we are targeting a population that where we want some decline in the placebo to show that there is less decline in the treated, this would be the ideal population. And then there is a final time when there is a very quick decline, when these children may lose ambulation very, very rapidly. So it's really important that if, as we see in many of the drugs we are using now, when the aim or the possible effect is to slow down deterioration, this would be the ideal population. So it's not a matter of which measure we use, but which population we select. And so this is ideal for this population. We want to avoid to put these children in the placebo because we wouldn't see any progression, and we want to avoid to take these other ones because there is a very fast deterioration, and so they may lose ambulation very quickly soon after we start a possible treatment. And so there has been an effort to identify, can we identify with the available scales the children who are likely to lose ambulation? And so we can do this with a 6-minute walk test, we can do it with the North Star, we can do it with the time to rise off the floor. Another element is the duration of the study. This is something we have learned a lot in the last few years. Several studies have shown a very clear trend, but they didn't reach significance in the first year. But when we look at the long-term results, we can see better results. These are the results with dystrophin restoration drugs, and these are the results, the first results on the eteplirsen, where in the first year there was practically no difference, but with increasing time there was a much more evident difference with external controls. And the same applies to other exon-skipping drugs, and also with the Translarna. I think Laurent tomorrow will tell you more about Translarna and all these drugs, so I will go very quickly. While in the first year there was a trend trend, which was not always statistically significant. When we follow these patients in a registry, that when we look at the long-term results compared to with propensity matching with an external population from Synergy, with increasing time, there was a delay in loss of ambulation, which was over 3 years. So this is much more significant than 3 points and so on. And even with other drugs like Givinostat, which has a different mechanism of action, doesn't go into dystrophin restoration, if we had stopped the trial at 12 months, we would have not seen a statistical significance, which was obvious at 12 months. So what is the message? What are the conclusions? Is that we have been obsessed with outcome measures. I have probably spent the last 15 years developing, revising outcome measures, but the truth is that we cannot blame the outcome measures. We cannot just think that one outcome measures will be able to address all the different cohorts and duration of the study. So the lesson we learn is that really we need really to think of trial design rather than outcome measures, and the choice of the outcome measures must depend on the particular trial. We should not start from the measure, but we should look at the inclusion criteria with all the things we discuss, age, functional level, and so on, but especially on the mechanism of action of the proposed drug. Because if many of the drugs we are using now in Duchenne are delaying progression, I think it's important to target even more the right population. And the duration of the study is something more. Where are we going? What is the final conclusion? Is that we should look at these outcome measures. We should put into context all the things I have discussed, but we should also think of trial designs which have a different way. Today, in the first sessions, we were discussing about the possibility of synthetic data as external controls, especially if there is a long-term duration. We may not be able to have placebo-controlled trial to show the efficacy. So we have to think in a more structured way, not just focusing on one outcome measures. And recently, in the recent studies, using MRIs or activity monitors have been extremely useful to have additional data. They may not reflect all the activities of daily living that we know are meaningful to patients, but are extremely important to provide concomitant data. I wouldn't even say additional, but concomitant data. So I think we have to start thinking in a different way. So outcome measure is not the major issue, but is trial design. And I think this was my last slides, and so thank you for the attention.
Any questions? So you partially answered some of mine throughout your presentation, but do you think we should be using composite measures as a primary endpoint, or do you think we should be using a single test that might be more sensitive to change for the specific population?
This is the question I didn't want to hear, because I started saying we shouldn't discuss outcome measures without knowing which population we are supposed to use it for. So I refuse to answer the question. No, in general terms, I think if we are, because we have instructions from regulators that very often regulators or the companies wants to go for wider population. So the idea, if we are including children from the age of 4 to 12, then composite would be great. In SMA, we are actually developing composite scores which are working very well. So I think this could be an answer if the population was, it's either we go for a proof of concept in a homogeneous population where a single scale measure would be better, but if we are forced to have a wider population, then composite scores, of course, would be a possible answer.
Wonderful. Any other questions for our speaker? All right.
Okay, thank you.
A few announcements before lunch. If you did leave your poster last night, please pick it up in the Tiffany room. Otherwise, it will be disposed of after lunch. And then we also need slides from Dr. Hare. All right, and now we can go to lunch. Thank you.
Eu quis amar, mas tive medo. Eu quis salvar meu coração, mas o amor sabe um segredo. O medo pode matar o teu coração. Água de beber, camara. Água de beber, camara. Eu nunca fiz coisa tão certa. Entrei pra escola do perdão. A minha casa vive aberta. Abri todas as portas do coração. Água de beber, camara. Água de beber, camara. Eu quis amar, mas tive medo. Eu quis salvar meu coração, mas o amor sabe um segredo. O medo pode matar o teu coração. Água de beber, camarada. Água de beber, camarada. Eu nunca fiz coisa tão certa. Entrei pra escola do perdão. A minha casa vive aberta. Abri todas as portas do coração. Água de beber, camarada. Água de beber, camarada.
sapore eterno, resta quel che resta. E se qualcosa poi non va, senti quel vuoto dentro. Le strade non hanno un nome e non sei contento, non sei più contento di te. Che cerchi me in questo tempo che passa, mi accorgo che mi manchi. I miss you, oh no, I miss you. E se qualcuno ti dirà che si può anche impazzire senza discutere, spiegare al tuo cuore, resta quel che resta. E se qualcosa ci sarà, oltre quegli occhi chiusi, devi pretendere una vita migliore, essere contento di te, che ami me in questo tempo che passa, mi accorgo che mi manchi. I miss you, I miss you. I miss you. Miss you. Essere contento di te, che ami me in questo tempo che passa, mi accorgo che mi manchi. I miss you, I miss you. Bella pure questa.
I saw you last night and got that old feeling. When you came inside, I got that old feeling. The moment that you danced by, I felt a thrill. And when you caught my eye, my heart stood still. Once again I seemed to feel that old yearning, and I knew the spark of love was still burning. There'll be no new romance for me, it's foolish to start, for that old feeling is still in my heart. there'll be no new romance for me. It's foolish to start, for that old feeling is still in my heart.
You get a shiver in the dark, it's raining in the park, but meantime, sound of the river, you stop and you hold everything. A band is blowing Dixie, double fall time. You feel alright when you hear the music ring. Well, now you step inside, but you don't see too many faces. Coming in out of the rain, they hear the jazz go down. Competition in other places. But the horns, they blow in that sound. Way on down south. Way on down south, London town. You check out Guitar George, he knows all the chords. But it's strictly rhythm, he doesn't want to make it cry or sing. If an old guitar is all he can afford, when he gets up under the lights to play his thing. And Harry doesn't mind if he doesn't make the scene. He's got a daytime job, he's doing alright. He can play the honky tonk like anything, saving it up Friday night with the Sultans, with the Sultans of Swing. And a crowd of young boys, they're fooling around in the corner, drunk and dressed in their best brown baggies in their platform soles. They don't give a damn about any trumpet playing band. It ain't what they call rock and roll. And the Sultans, yeah, the Sultans, they play Creole. Creole. one more thing. the Sultans. We are the Sultans of swing.
Is it?
Yes.
Afternoon. Is it on? Okay. Afternoon, everybody. Hope you had a good lunch. We are running a little bit late. Still people are coming in, but I guess we need to move on. We'll reduce the upcoming break here by 5 minutes, so you only have 10 minutes. But it's a very early break. And it's my pleasure to be here with Paloma Perez to chair this session. Obviously, I'm not Renata Knox. I'm trying to make up for not showing up for my chairmanship yesterday. And this session will be on new phenotypes. And the first speaker is Michael Hare, who will be presenting new phenotypes in myasthenia gravis.
Well, thank you so much to the NMSG and the committee for inviting me to talk today, and also for giving me this really hard topic. I hope that I can say something that people here will find important today. I'm going to be talking about new phenotypes and classifications of myasthenia. These are my disclosures. I work for companies that produce treatments for myasthenia. So we're going to talk a little bit about some of the traditional classifications of myasthenia, and then I'm going to talk a little bit about what we might consider new phenotypes and classifications. And I'm intending this talk to be something that will get people to sort of think about how we move forward. So myasthenia, traditionally, the way that we've tended to classify it is in a number of different ways. So we think about generalized, and here. How's that? Better. Okay. We think about ocular versus generalized disease, whether or not we're going to give symptomatic or immune therapy based on whether it's generalized or ocular, thymectomy or not. And then the other thing we think about is age of onset. Is it an early onset person, a premenopausal woman, a postmenopausal woman? Or is it very late onset, people older than 65? And there's different phenotypes that may present in these different age categories. And then, we always think about antibody status because it affects our therapy choices and whether or not we're going to do a thymectomy. But we're going to depart from these for a little while today, and I hope I can sell you on the treated patient as a potential new classification for myasthenia. So this is where we're at in 2025. So many choices of therapies approved internationally for patients with myasthenia. And personally, I feel like that guy up there, and I think all my patients feel like that guy up there when I'm trying to think about what I'm gonna prescribe for any one of my patients. And we have a whole host of immunosuppressant therapies. We have FcRn therapies that remove antibodies. We have C5 complement inhibitors and IVIG and plasma exchange, as well as thymectomy. And then there's just so much coming. There's so much at this meeting. There's a bunch of speakers in the session later today. There were a number of posters last night talking about all these wonderful therapies that are coming. We have CAR T therapy, different B-cell depleters, cytokine inhibitors, JAK kinase inhibitors, C1 complement inhibitors, so many things coming, and they all potentially are going to work on the order of weeks to months for our patients. So how are we doing? So, very old data from earlier this century and late last century shows that mortality from myasthenia is low, like 5%. Our group published a study last year that was looking at mortality in Medicare patients, so patients over 65 in the United States. And our patients, although the mortality rate in myasthenia doesn't seem to be going up, patients with myasthenia in the 65 to 80 category have a slightly higher mortality rate than their age-matched counterparts with similar comorbidities. So that begs the question is it something we're doing? Is it related to the disease? Is it related to the treatments? We don't really know. Once patients are over 80, the myasthenia patients are actually living a bit longer than their counterparts. And I always, when I'm thinking about treatments for myasthenia, I always kind of come back to the consensus guidance where we talk about, a good clinical status for our patients is achieving no symptoms with minimal side effects. I recently had the opportunity to speak with one of my colleagues at a patient meeting a couple of weeks ago, and this is what she said. Amanda Guidon said, be ambitious. And I think that's really should be all of our goal when we're thinking about patients with myasthenia. So how are we doing? So we are not doing great. So if you look here, I have on the left side of the slide what would be considered traditional treatments for myasthenia. And you can see that the minimal manifestations or better rate in those, with those treatments is somewhere around 60 to 70% of patients. On the right side of the slide, I have what would be considered newer generation, or what a lot of people call targeted therapies. And you can see that the rate of minimal manifestations is not quite as high as some of the traditional agents. One of the advantages of some of the newer therapies, though, is that the time to achieve that status may be faster than some of the traditional therapies. So why is our rate of response not good? And this is what I was thinking about. So is our immune system target the right one? Is it the right one to modify the immune system? Is it the correct immune target for your particular patient? Are we using enough concurrent therapy with our patients, or are there shifting immune phenotypes over time? So just to kind of orient us, we all know this, but at the neuromuscular junction, the pathology of myasthenia, there's sort of three main things that are going on at any one time. So with acetylcholine receptor binding patients, there is antibody blockade, number one, where you're blocking the acetylcholine receptor. There is number two, where we get endocytosis of acetylcholine receptors off the surface of the muscle. And then there's number three, activation of the complement cascade and destruction of the neuromuscular junction. So what about incorrect immune targets? So the group at Yale, Kevin O'Connor's group, published this paper last year, which was really cool. They analyzed the data from the randomized rituximab trial in acetylcholine myasthenia, and they were looking at antibody types. And with the antibody types, which of those mechanisms were those antibody types, the epitypes of these antibodies, able to do? And you can see that the majority of the antibodies were activating complement. About 30% of the antibodies were blocking the receptors, and about 60% were internalizing the receptors. But if you look at that together, almost 90% of the antibodies were doing multiple things. So how does that, play when we're thinking about immune system therapies? So, if it's activating complement alone, maybe all they need is a complement inhibitor. If it's multiple mechanisms, maybe they need multiple treatments that span different strategies. Or if it's just blocking, can we just pull out the antibodies? So thinking about B cells, I'm going to talk a little bit about B cells and T cells and how we're thinking about it. So this version of this diagram was put up yesterday, thinking about where CD20 and CD19 is represented on B cells. So for anti-MuSK myasthenia, we think about short-lived plasma cells. For acetylcholine myasthenia, we're thinking about long-lived plasma cells. In late onset myasthenia, there's a lot more CD20 cells. So for MuSK and for late onset MG, it may make sense to use CD20 blockers like rituximab. But for acetylcholine receptor myasthenia, it may be important to be hitting the pre-B cells or the immature B cells, in which case maybe CD19 is a better target for these patients. And there's some trials that suggest that this may be the case. So here's the BEAT-MG trial, which was published in 2022, and the primary endpoint was the ability to wean prednisone in patients treated with rituximab who were acetylcholine antibody positive, and they didn't hit the primary endpoint. There was no difference with treatment. However, we did this study a number of years ago where we looked at the effects of rituximab in MuSK myasthenia at multiple centers, and we saw that 70% of our patients achieved minimal manifestations compared to 30% of the patients who did not receive rituximab. And I think everybody in this room knows that rituximab is a really good therapy for MuSK myasthenia, but this is suggesting that maybe it's the type of B cells that we're hitting that could be the problem. And just this past year, oh, that's mislabeled, sorry. This is an anti-CD19 antibody in inebilizumab, and this was in acetylcholine receptor patients, and they showed a difference at about 6 months in patients treated with a CD19 medication compared to placebo, which is different than the BEAT-MG trial. So maybe it is the type of B cells that we're targeting. The new kid on the block is CAR T therapy, and this is all directed at B cells. So CAR T cells are designed where a patient's own T cells are educated with either DNA or with RNA, and then given back to the patient to exert effect on a particular condition. These were designed to treat cancer. A is the method that's used to treat cancer, and the idea is by using a DNA-based strategy, once the CAR T is put into a patient's body, it continues to replicate and fight off the malignancy. The problem with this strategy is that as these cells are replicating, we get the bad side effects that all of us as neurologists are called to see in the hospital, with immune system reactivity that can affect the central nervous system or a cytokine release syndrome. One of the strategies that's being used in myasthenia is to use an mRNA version of CAR T, and the advantage of that is that the RNA degrades over time and we think would cause less of those bad side effects, but may be helpful. So in myasthenia, one version of CAR T is targeting the BCMA receptor, which is an early plasma cell marker. And in this trial, what they showed is treating with this BCMA CAR T therapy resulted in a significant decline in all MG metrics over time that was sustained for up to a year, and in some patients longer. In patients who started to relapse at about a year, you could be retreated with this and then again return to a better clinical status. So again, thinking about B-cell target seems to be important. T cells are also really important in myasthenia. What's going on there? So there was this nice paper published last year by our colleagues in Sweden that was talking about the types of T cells or difference in T cells between myasthenia patients and controls. And what you see on the left is that at the resting state, patients with myasthenia in red had a higher activity level compared to healthy controls, and that was true for CD4 and CD8 cells. But then when the immune system became active, it was the myasthenia T cells seemed to be fatigued in some way, and they didn't rise to the same level as somebody who was a healthy control. So there may be some difference in how those things are happening that may impact the disease status in myasthenia. And we know from old studies of azathioprine and mycophenolate that if we target B and T cells together, that our patients do really well. So on the left, this is the pivotal azathioprine trial, which showed that patients with the solid line were less likely to relapse once they had achieved remission. And on the other side is a retrospective study from multiple centers that showed that at about 12 to 14 months, patients who were treated with CellCept monotherapy and with prednisone achieved minimal manifestation status or better. So T cells seem to be important and something that we should be thinking about in our patients. Which begs the question of, when we're thinking about our patients, should we be talking about immune modification or treatment? And on the left side of the slide, these are things that are targeting the B and T cells themselves. So we have steroids or non-steroidal immunosuppressants, we have thymectomy, and then there's the things that are acting sort of on antibodies themselves, like FcRn therapies or complement that might be considered treatment, but maybe not immune modification. And how does that play out? So if we look at FcRn inhibitors, it's pretty reliable that patients will get better with myasthenia when they're actively being treated with these medications, but once the treatment washes out of the system, patients' antibody levels rise and their disease status gets worse pretty reliably. So to get around that, you could just treat a patient persistently with an FcRn inhibitor, and their clinical status will stay improved, but they need to be on that as a chronic therapy. And I would consider this to be treatment rather than immune modification. Versus thymectomy, which might be considered immune modification. And when patients in the MGTX randomized trial of open thymectomy and acetylcholine receptor myasthenia, about 40 to 60% of the patients were in minimal manifestations and were able to come off prednisone completely over time at about the 3-year mark. Some of those patients, as they came off prednisone, lost their minimal manifestation status, but if they went back on treatment, they were able to get back to that good clinical status. And that suggests that the surgery itself is modifying the immune system and maybe treating the underlying disease by switching the immune system. And how does that play out with some of the traditional therapies? So there's 3 studies that I know of that looked at whether or not we can taper immune therapies in patients who are not being treated with any of the newer agents. And in about 60 or 70% of patients with mycophenolate or with azathioprine, we can wean drug and keep patients in minimal manifestation status. So it seems that we're impacting the immune system in some longer standing way if people are able to get on less drug over time and maintain that good clinical status. So my mantra, my friends from Vermont who are here with me know that I'd say this to our residents all the time. I always tell them to think. And I think in myasthenia, some of the things that are starting to perpetuate in the community are that new treatments are safer and more effective than traditional ones. And people call the newer treatments targeted therapy, and people are starting to say, well, those are better than traditional imprecise therapies like CellCept and mycophenolate and azathioprine. And prednisone is the only medication that causes meaningful side effects in myasthenia. And I couldn't resist putting this in. So I designed a unit to look at side effect burden in myasthenia with a bunch of colleagues. And we had a prospective study that we did at UVM where we looked at patients and their side effect burden over time, and a higher score on the adverse event unit is suggesting a higher burden of side effects. And you can see here, we did this before we had FcRn inhibitors and before we had any other complement inhibitors other than eculizumab. But patients who were treated with low-dose prednisone and patients who were treated with mycophenolate seemed to have less of a side effect burden than the patients who were getting eculizumab. Food for thought, more coming, I hope. MG patients have varied immune targets. Markers of individual patients' MG are needed. So can we figure out a way to tell this patient you need a B-cell inhibitor and this patient you need a T-cell inhibitor? And I think we need comparative effectiveness and comparative side effect burden trials in myasthenia now that there's so many options. So I'm going to shift quick to a second new classification of myasthenia. So early versus late myasthenia. So the traditional way we're thinking about early versus late myasthenia is the age of onset. So early versus late versus very late. And the big difference pathologically among these three categories is that the patients over 65 are less likely to have thymic hyperplasia, but are more likely to have a thymoma or thymic atrophy. They're also more likely to be acetylcholine receptor antibody positive. And the other thing that we know about these categories is that the patients that are presenting earlier in life tend to be harder to get into a minimal manifestation status over time. So purple, blue, and orange are indicating patients who are still symptomatic from disease after treatment. But what I want to try and sell you on today is proximity to symptom onset as a new early versus late idea in myasthenia. And what do we know about that? So this is a very famous paper by Dr. Grobe, probably one of the most cited papers in myasthenia. And this is what we tell our patients, right? So you will likely have the worst amount of disease you will ever have in the first 2 years that you're symptomatic from MG, which suggests that there's something important happening during those 2 years. The Austrian groups published this recently, 5 years ago, which showed that it may be even just within the first year that people tend to hit their nadir, suggesting that this is prime time to be thinking about treatment for our patients. And how does that play out? So I wanted to call out the BEAT MG trial again. So these were patients that had disease for 5 to 7 years before they received rituximab, versus the Rhinomax trial, which was published a number of years ago out of Sweden, which showed benefit for rituximab in patients with acetylcholine receptor disease. The difference was that those patients were treated at 4.5 months after symptom onset. And what did we find? So the patients in the Rhinomax trial achieved minimal manifestations status at 24, 36, and 48 months after treatment, which is quite a good response to therapy. And a follow-up study was done in Sweden. And again, it showed that if patients were treated at less than 6 months of their treatment onset, their time to remission was only 6 months compared to patients who had been symptomatic for longer than 12 months. Their status didn't change until about 16 months. So treating early seems to be important. And then it's hard to know what's going on, but there's been some really interesting data coming out of the Yale lab about what's happening to antibodies over time. And I believe there's going to be some more publications coming from this work later this year or early next year. But what they did is they took 50 patients from the rituximab trial, and they had 210 longitudinal serum samples. And what their data is showing there on the graph is that the epitopes of those antibodies are changing over time. So sometimes they're activating complement, at other times they don't seem to be activating complement in the same patient over time with treatment. So the immune system is sort of shifting what it's doing and maybe shifting the target for us to be treating our patients. And we've all had patients who are fluctuating after they've started therapy, where they're responding to something and then they start not to respond to that therapy and we have to switch to the next thing. And what is really going on with our patient? And this may be one explanation for what's going on with them. It also suggests that if the targets are changing over time, that it may be important to hit those targets early and keep the immune system suppressed to sort of modulate the immune system before this shifting starts to happen. And I think people are starting to think about, timing of rituximab based on those studies that I just showed you. So I think age of onset is related to disease severity, but I think aggressive early immunotherapy may also predict good outcomes in our patients. And the duration of myasthenia prior to treatment is important, and we should be considering that when we're thinking about our patients. So I hope that I've raised some questions for all of you thinking about where we go with myasthenia in the future, thinking about what are the best therapies or combinations of therapies to achieve a good clinical status. We need ways to predict what our immune targets are for any particular patient. When are the optimal times to be intervening? What is happening to our immune system over time? And can we predict which patients may not need ongoing therapy based on some of the things we discussed today? So I thank you for your attention and for putting up with that. Appreciate it.
Thanks, Michael, for this tour de force of treatment mostly, also a little bit on phenotype. Any questions? We have time for one or two. Yes?
That was a great talk, thank you. Could you comment about antibody-positive ocular myasthenia just for a bit, how you think about that, especially in terms of this idea about treating early and aggressively?
Yeah, so in the interest of time, I took out some of the slides around ocular MG. If you could pull my slides up, I still have them on there. I could shift ahead. Yes, so antibody positivity and ocular myasthenia, I think it's a 2.5 to 3-fold increased risk of generalizing over time. Yeah, I don't know if I'll be able to get there. That's okay. But if you treat patients with ocular myasthenia with immunotherapies, there's old data to suggest that they won't generalize. And there's also data that if you do that earlier in the course of their disease, they're less likely to generalize. So it's a really important question. What do you mean by habits?
Meaning, like, if they're feeling better, maybe they're doing more physical activities, maybe they're out of work now, they're returning to work, or there are other stressors in their life that come about.
Yeah, hard to say. I mean, it's interesting to think about that. It'd be interesting to study that. I think there's some good data. There was data presented last night on exercise in patients with myasthenia at the poster session that suggests that that's associated with good outcomes. The Swedish groups have a lot of data on dedicated exercise programs and needing less medication to achieve good clinical status. So I think exercise is really important and probably needs a lot more study.
Thank you, Michael.
Thank you so much.
Okay, so now let's move on to our next speaker, Dr. Johanna Hamel. The title changed a little bit, so she's going to talk about distance learning, distance research in myotonic dystrophy type 1.
All right. Thank you to the organizing committee to give me the opportunity to talk about my work, which will be about distance learning, distance research, and just to comply with the assignment remote phenotyping, but I don't like that word, so I changed the title. These are my disclosures. So the talk will have two portions. I'll talk about distance learning, distance research, which will include my study remote assessments and genetic analysis in DM1, REACH-DM. But then I will shift gears and talk about a smooth muscle phenotype, smooth muscle disease, and we'll talk about the women's health study and sexual and menstrual function. So starting with the first topic and the problem. In DM1 in the US, people with this common but rare disease are dispersed geographically, and so are the research sites. Travel is costly and burdensome. I don't have to tell this group how myotonic dystrophy affects whole families, and usually there's one in each generation affected, if not more, with a tendency to get worse from generation to generation. There's day-to-day variability in DM1 symptoms, and so it poses the question whether it's a risk to rely on a single point determination of a measurement, for example, myotonia, which is a very fluctuating symptom. So the question is, can remote assessments be done to overcome some of those barriers that I just mentioned in people with DM1? And if they can be done, will they be useful? So to answer that question, I followed a development path of remote assessments over the last couple of years, and I'll walk you through that path by showing my data. And we'll start with the feasibility. Is it doable in this population? Moving on to, yes, it's doable, but what are these efforts and measurements? Are they worth it? How noisy are they when we repeat them in the remote setting in consecutive remote study visits? And then lastly, we'll walk you through comparison to what's considered the gold standard, in-person, on-site obtained measurements. Starting with the feasibility, this idea and project launched before the pandemic by us putting up a toolkit, putting in some devices for quantified assessments in the home environment, including a tablet so that people could connect with their home Wi-Fi on a one-on-one Zoom visit with me, and me doing some remote study visits. And that worked well in 23 people, and because it was fun, I expanded the study by putting more stuff into the toolkit and sending it out to patients and conducting more remote study visits. And the way this works is that I connect with people on a one-on-one Zoom. I conduct a medical and symptom onset interview, followed by functional assessments, which I will go over on the next slide, followed by people filling out surveys on their tablet at a convenient time for them, patient-reported outcomes, which are listed here on the right. And then people wear an activity monitor for 7 days on their own, obviously, and then fill out an exercise survey at the end of the 7 days. And then they go out in their community and have their blood drawn, and then they ship that back to us. And these are the assessments. So on the left-hand side, you see assessments of the upper extremity function, the 9-hole peg test, grip strength, pinch grip. On the bottom left, you see the VHOT, video hand opening time. We do the VHOT a little different than usual. We do a VHOT normal, standard VHOT after resting time, but then we do a back-to-back VHOT to assess the warm-up phenomenon, which I think is meaningful to patients to quantify it. And then in the middle, you see lower extremity function with the timed up and go and the 10-meter walk. On the right-hand side, you see oropharyngeal assessments, tongue and buccal strength. And then we assess respiratory function by checking FVC and SNP. I also do a 6-lead EKG. We have added in cognitive testing, a 5-time sit-to-stand, and a speech recording. And just one highlight here is that we video record these assessments. So we have videos of the 9-hole peg, timed up and go, 10-meter walk, VHOT, 5 sit-to-stand, and speech recordings. So we have accumulated hundreds and hundreds of videos that are sitting in my box folder. And so just to go over the workflow again, we recruit patients remotely through the registry and other means. We ship out the toolkit. We have a remote study visit. We have them do questionnaires where the activity monitor, remote blood collection. The toolkit is returned to us. We recycle, clean it, and then it goes to the next person. And all this magic is happening because of Aaron Richardson. And so where are we at the moment? Obviously, the study is ongoing. I have enrolled 89 people for initial remote study visits, enrolled from 25 states, which includes Alaska, which was a lot of fun. And then we have 12 families where there was more than one participant from that family. And if I include the REACH DM Kids study that we launched as well, including the whole spectrum of live time, so patients from 0 to 18, if I count that in, then we actually have 25 families at times spanning 3 generations. Slightly more females, the age range is wide, but what I want to highlight here is that research-naive patients make up 47%, so people that tell us that they've never participated in a research visit before. So we were able to engage those folks as well. 23% needed some assistance from another person in the household. These are just some feasibility completion rates, so how many people were able to perform the assessment in the home environment. It's looking pretty good for the different study visits. So in the orange, you see the initial, then we did reliability visits, so it was consecutive remote study visits, and then on-site study visits. And then I also started to do annual visits and just connect with folks again after a year. but I will not show more of that data today. And just one thing I want to highlight, because safety is always the priority. The supine spirometry are a little lower, and that's because if people have trouble getting up, there's no one in the household to get them up, or they have trouble breathing lying flat by history, I don't do the assessment because I'm not there to help them. And then, so the expanded study also included the activity monitors, and that's looking pretty good. I think our success is increasing because we're getting better at how to counsel patients and ship it out on time, etc. Either way, particularly the wrist-worn one is worn sufficiently by most people to capture sleep and activity. And the waist one, I let them take off at night, so it's usually worn during the day, so that's why the time worn is a little lower. What do patients say? Some people say that they have trouble with the technology, a few here on the left, but most people navigate it fine, maybe with some help, as I mentioned. And just to highlight, some people are not used to use tablets, but yet did fine with the study. And when we asked them, would you prefer to do a remote on-site or either study visit, 31% would prefer a remote study visit in the future. In terms of blood return, 91% of our participants have returned their blood to us, and we actually return the genetic research test result with their repeat length to folks in a letter accompanied with interpretation of the results, and we did that in 95% already of the analyzed samples. So feasibility is looking pretty good, but is it worth it? What are these measures really like in terms of noisiness when you repeat them? Are they all over the place? Obviously, it's a less standardized setting. So we completed consecutive remote study visits, repeating the functional assessments within 3 months' time. This work is also the work of Laura, who helped a lot with the analysis and spent 6 months with us in Rochester. Here are the patient characteristics of this sub-cohort of 40 people, just highlighting wide age range, wide range of disease severity, and the median days between each study visit was 25 days. And so here's the data. It's the ICCs here on the right side. In green, color-coded are excellent scores, so really excellent reliability of the measurements. And then in blue, good, and yellow, moderate, the VHOT, the first VHOT after resting time is on the bottom, not surprisingly to us, because it's a fluctuating symptom, but the functional measurements are really up there. And what I'm also happy about is the spirometry that's really holding up. The VHOT, the warm-up VHOT is a little more reliable, and that is because the greater the myotonia, the greater the variability. And so because with the warm-up, the myotonia gets better, the less variability. Now, tongues are tough, but tongues are important in DM. And that's because it has high DMPK expression, and it causes a great deal of disability in terms of dysarthria, dysphagia, and they're not always well measured. But I'm happy about this result in the test retest reliability that we're able to capture this symptom. So in our interval summary here, remote assessments are reliable, they're safe, we didn't have any falls, well perceived by participants, and what I'm particularly happy about, we are able to engage folks that really usually don't participate in research. That includes doing study visits after hours, because people are at work during the day, at least those participants. So moving on, what does this, a lot of questions I get, well, how does this hold up to the gold standard, the on-site assessments? So we did a sub-study including 30 people that actually came to our research center, and here's again the characteristics, broad range of age, a little more women, but a broad range of disease severity in terms of age of onset and repeat length. And so what does the reliability look like? A little lower than what you just saw, but still holding up, particularly the functional assessments on the top there with spirometry, the winner, and then hand grip, pinch, 10-meter walk, and then again, as expected, the VHOT is holding up in the moderate range. And so I just wanted to mention here that with the VHOT, we exclude, that's why the numbers are a little lower, we exclude people who do not have clinical or subjective myotonia in the ICC, because that would bring up the ICC and make it look better, but it's not really useful because, you know, if someone doesn't really have myotonia, why would we want to keep measuring it? So why is there a little bit lower reliability maybe on-site? Well, there are different sources. So the location, the research space obviously has a different floor. You know, it might be slippery, it might be grippy, whatever it is. At home, I can just do it in the same place. Evaluators, there's usually more than one evaluator at a site. Circumstances are variable. At home, I can tell them to wear exactly the same clothing, the same shoes, and the patients are able to keep the temperature at whatever they like at home, while in the research center, if it's freezing, can't do much about it, but it's definitely going to impact myotonia and possibly strength testing. So next steps are to increase our enrollment, and to everyone here in the audience, referrals welcome in the US. We want to expand our outreach. We have added additional outcomes, and we're just working towards automated analysis, AI-driven analysis of our video-based functional assessments to make use of these hundreds of videos. And so why am I keeping doing this? Well, I want to assemble a large cohort really representing the spectrum of DM1, hopefully contributing to a genome-wide association study in the future. But I also want to get ready for the future when we have several therapies on the market, and we have to figure out which one works for which person, which one works best for which symptom, and what if there's a plateau? How do we make treatment decisions, when to switch, for example, from one drug to another? Those studies need to be done at low cost and really utilizing the community. And so that's what I'm hoping to do in the future with this. So shifting gears to the smooth muscle phenotype, that is an idea that came out of the clinic, and we all know that the smooth muscle is understudied and under-recognized in DM. And so in the clinic, I happened to learn from patients that they had trouble with sex or trouble with menstruation and lots of pain that made them stay in the house. And so I don't know much about this topic, and a lot of those questions usually the OBs get to hear and not us. And we usually don't ask these questions either, our patients. So I got some help by the specialist, so a person who's focusing on sexual function in several diseases, and the director of the Maternity and Fertility Clinic and High-Risk Pregnancy in Rochester, and statistical support, and my partner in crime, Jeannie Dectebron. And so this study was based on the fact that we thought, well, these muscles are probably affected. And yes, we know from delivery and pregnancy studies that there is a problem sometimes with reproduction, but you know, women's health is more than reproduction, and so there is possibly problems with sexual and menstrual health because of smooth muscle disease. And so our hypothesis was that there's probably greater pelvic floor and menstrual dysfunction in DM compared to FSHD due to smooth muscle involvement. So we created a large survey, which I will only provide glimpses of today. It had a ton of intimate questions, and it was not anonymous. So I'm very happy about the return. 106 DM1, 68 DM2, and 109 FSHD patients responded. And here you see that the DM2 patients and the FSH patients were a little older, not unexpected. The age of onset was older in DM2 compared to FSH DM1, as expected, and the diagnostic delay was greater in DM2, as expected. By the way, there were no differences in all these factors down below, which are important, like ethnicity, relationship status, marital status, disability status, and so on. So here I just want to give a glimpse of the data that we started to analyze. And so, for example, we created a survey asking whether any of those DM symptoms impacted their ability to engage in sexual activities. And they were able to select as many as they wanted. And so what this showed, in particular here, and the only statistically significant differences between DM1 in blue and DM2 in yellow are that the DM1 women particularly reported, over 70% reported that excessive daytime sleepiness and muscle fatigue impacts their ability to engage in sexual activities. More, the DM2 folks reported that proximal lower extremity weakness impacts their ability to engage, which is, again, expected. that difference. And then I was interested in the GI issues, and there is a decent amount of people, up to 30%, who also reported that GI symptoms impact their ability. Now, we included the pelvic floor distress inventory, PFDI. This is a score where a greater score indicates greater dysfunction on the y-axis, and here color-coded the different diseases. You see that DM1 and DM2 have greater urinary pelvic floor dysfunction, greater pelvic organ prolapse dysfunction, for example, vaginal prolapse. There was no difference between the groups in the colorectal anal distress inventory. And the total pelvic floor function index was also different, so greater dysfunction in DM1 and 2 compared to FSHD. Now, just giving a glimpse of some of the data from the menstrual health, there was no group differences in terms of menopausal status, parity, menstrual flow, interesting, not differently. But we did see slight differences here in pelvic pain, so the DMs both had more pelvic pain compared to FSHD, and then there was a little more lower back pain with the period, I should have mentioned, with period, these symptoms in the DM1 group. And then another thing we're just exploring, this is not statistically significant, but it was interesting to me that DM patients seem to use more pads compared to FSHD there. Moving on, this is the question, those are OB/GYN questions that they give out in clinic, for example, by the way. So we derived those surveys, we created the survey based on tools that I used in the OB world. So how often did you sleep with a tampon in place at night? Interestingly, the DM1s, 55% reported that they never do that, which is in discrepancy to DM1, DM2, and FSHD. And so the question that I have is that because the muscles are weak and they can't hold it and it comes out, or is it just personal preference, which would be strange? But we also gave participants the option to give us free text answers and ideas in the survey, and reading through all those text messages, I have a suspicion that there's a good reason why they're not using the tampon, because their muscles might not hold it. In general, though, there was no difference in tampon use during the day. So the conclusions here are that participants with DM1 and DM2 report greater pelvic floor dysfunction compared to FSHD, possibly attributed to the preferentially affected muscles in DM, and that DM symptoms impact engagement in sexual function. And here, muscle fatigue and excessive daytime sleepiness really high up on the list. And so more research is needed to better understand the biological basis of smooth muscle dysfunction, particularly in the female organs, which I'm interested in pursuing. And then I think some qualitative work and outcome measure development is needed, because none of these symptoms are currently assessed in clinical trials, and the drugs that we're administering to patients should be targeting these muscles as well. But if we don't ask the questions, we won't know. And with that, I want to thank all participants and their families and friends, and the team, Erin, Jeannie, Laura, and Eleanor, and Chandani in the Thornton lab, who does all the repeat analysis. And I'll take questions. Thank you.
Any questions from the audience?
Hi, thanks for the great talk. So most of the outcome measures are currently used for the DM1 natural history studies. So I was wondering, in light of the correlation between the in-person and remote assessment, do you think the remote assessment might have a role in the natural history studies going forward, so that we can kind of optimize and reduce the time of the in-person physical assessment?
Just to make sure I understood the question right, so are you wondering if the remote assessments might be useful to supplement natural history studies or even clinical trials in the future?
Or to conduct the natural history studies remotely rather than...
this is sort of a natural history study. I have larger goals because we're also trying to develop new outcome measures that way with our videos. But really, yes, because I do think our research studies at the moment, our natural history studies, are only capturing a fraction. The NTM study certainly has the broadest inclusion criteria and has really captured a lot of people, but just from doing the remote research, I've been reaching people that really would never even consider doing a natural history study because their DM isn't much of their life. They're even working or just disengaged or geographically disadvantaged to participate. So I think with this study, one of the opportunities is really to capture the full broad spectrum. We also know that the DM mutation in the US occurs at about 1 to 2,100, which is much higher than we previously thought. This is based on the New York State prevalence study. And so there are a lot more people out there in the community that are not studied in research studies. Maybe they're in the clinics, but maybe not even in the clinics. So my goal is to ascertain full families, really maybe even diagnose in the future by including non-diagnosed at-risk patients in the study, and really to achieve full ascertainment, which I think is important when we talk about GWAS studies.
Another question there.
Fantastic talk, thank you so much. Do you have any first data on the reliability of the wearable sensors when you use it 1 week for physical activity?
So the actigraph, we actually have only deployed at the initial visit and then a year later, so not at the, just a couple weeks later, so we won't have that data. I think that will require a little bit more work. I think there's also, we try to capture a week that's representative, but some, there is more variability. It's a lot of data, and we're just getting to the data set to really make sense of it. So I don't have that. I don't have an answer yet. Yep.
Another one there.
Hi there. Great to see the women's health aspect addressed. And sorry, I'm over here.
Yeah.
Yeah. One of the questions I have, and you've got a fantastic PT in your team, and you've already collaborated with the obs and gynae physicians. Is there a plan then to actually do the same collaboration when we're looking at possibilities for this group in terms of rehabilitation interventions that our women's health PTs are very good at?
Sorry, I didn't quite hear it. Do you mind just...
I'm normally quite loud. So it's great to see that you've collaborated across specialties with the obs and gynae physicians, and you've got a fantastic PT in your team who we know well. Is there an opportunity to expand that to actually look at rehabilitation interventions for these women that you've highlighted with these problems?
Oh, that would be wonderful. Yes, so we don't have plans that way. This is just the initial step, the survey. We would like to do qualitative work. We would also like, intervention work would be great. I send people to pelvic floor PT, for example, all the time, and not sure that we have a lot of data to support that or even get really strategies or inform the PTs that we ask to do this. So no, that's a great suggestion for the future, and always interested in collaboration. So, yeah.
Thank you very much, Dr. Hamel.
Then we will invite Dr. Jim Dick from the Mayo Clinic, who will be talking about vasculitis and neuropathies in large and small vessels.
Thank you very much. So I want to thank the organizers for inviting me here. Several years ago, the organization changed its name to the Neuromuscle Study Group, so I am excited to actually give you a talk about peripheral neuropathies. So I'm going to talk about the clinical features of peripheral neuropathies that occur in large arterial vasculitis compared to nerve microvasculitis. So we're going to look at the clinical features, the temporal evolution of large arterial vasculitis compared to microvasculitis, and look at disease associations, laboratory findings, etc. So all vasculitic neuropathies primarily involve small-sized blood vessels. and those are large arterioles or microvessels, because only small blood vessels supply peripheral nerves. Vasculitic neuropathies can be characterized as systemic with other organ system involvement, or as non-systemic, only confined to the peripheral nerve or muscles. Vasculitic neuropathies can further be subdivided into primary systemic vasculitis, no other disease associations, secondary systemic vasculitis with other disease associations, and non-systemic vasculitis. Vasculitis can be broken, vasculitic neuropathy in nerve can be broken down into large arterial, 75 to 300 micrometer, or nerve microvasculitis, less than 75 micrometer. So primary systemic vasculitis includes microscopic polyangiitis, eosinophilic granulomatosis with polyangiitis, granulomatosis with polyangiitis, mixed cryoglobulinemia, Henle-Schönlein purpura, medium vasculitis, poliaritis nodosum, and giant vessel vasculitis, which really doesn't occur in PNS. So connective tissues associated with nerve large arterial vasculitis include rheumatoid arthritis, systemic lupus erythematosus, Sjogren's syndrome, systemic sclerosis, dermatomyositis, mixed connective tissue diseases, sarcoidosis, Behçet's disease, infections, drug malignancies, inflammatory bowel disease. So non-systemic vasculitis often is a distal asymmetric neuropathy, non-diabetic lumbosacral radicular plexus neuropathy, Wartenberg's migratory sensory neuritis, and then there's the diabetic radicular plexus neuropathy, and localized cutaneous vasculitis. The neurologic symptoms and signs of non-systemic vasculitis are similar to systemic vasculitis, but are free of other organ involvement. One of the first descriptions of this was by Kernahan and Woltman at the Mayo Clinic in 1938. John Kissel and colleagues described an early series of non-systemic vasculitic neuropathy. My father, Peter Dick, coined the term non-systemic vasculitic neuropathy in 1987 in a series of 20 patients with nerve biopsy-confirmed vasculitis. Diabetic lumbosacral radicular plexus neuropathy historically has not been considered to be part of non-systemic vasculitic neuropathy. So, size of blood vessels involvement. Nerve biopsy is required to confirm the diagnosis of vasculitis, and suspected vasculitis is one of the most common reasons to do a nerve biopsy. To make a diagnosis of vasculitis, you need to have two features: vessel wall damage or destruction, and vessel wall inflammation. As I've mentioned, all vasculitic neuropathies involve small-sized blood vessels, and these include small arteries, large and small arterioles, capillaries, and venules. Small-sized blood vessels can be divided into large arterial vasculitis, 75 to 300 micrometer, versus microvessels, less than 75 micrometer. In the large arterial, you'll frequently have fibrinoid necrosis. In microvessels, you usually do not have fibrinoid necrosis. So here is an example of large arterial vasculitis, and you can see fibrinoid necrosis there. And on the right is a semi-thin epoxy section showing multifocal fiber loss typical of ischemic injury. In contrast, nerve microvasculitis does not usually have fibrinoid necrosis, but there's still infiltration of the vessel wall and destruction of the vessel wall. An autopsy study done by my father on 15,000 upper limb, 15,000 lower limb serial sections showed that the vasculitis occurs at all levels in the nerve. But the infarcts in the nerve start at the watershed zone of vascular supply. So in the mid-thigh level and the mid-arm level, you start to get central fascicular fiber loss, and then from there, it summates distally. And so this is the first study that really showed what the pathology of vasculitis was. And this he did this both in the upper and lower limb. So questions that we had are the clinical neuropathy patterns and temporal profile of neuropathy different between large arterial vasculitis and microvasculitis? Are systemic markers of vasculitis different between large arterial vasculitis and microvasculitis? Are associated diseases different between large arterial vasculitis and microvasculitis? And in our study, we took cases with confirmed vasculitis pathologically, and then we went backwards and looked at the clinical history and reviewed the charts. So all cases to go into our study had to be diagnostic or highly suggestive of of vasculitis. And this was done by Dr. Suntrapa, a fellow of ours who's from Thailand. So a total of 314 cases met our vasculitic neuropathy inclusion criteria. 36 of them were excluded due to insufficient clinical information. 278 cases fulfilled the final inclusion. Nerve large arterial vasculitis was 125 cases, 106 diagnostic, 19 highly suggestive. Nerve microvasculitis, 153 cases with 57 diagnostic and 96 highly suggestive. And the reason we have many more diagnostic and nerve large arterial vasculitis is that fibrinoid necrosis. So we wanted to be very conservative in how we're calling these, and it's a lot easier to be certain of that vasculitic diagnosis in large arterial vasculitis compared to microvasculitis. 52% were male, 88% were Caucasian, 2.5% were African American, 1.4% were Asian. The body mass index was higher in microvasculitis than large arterial vasculitis. Systemic autoimmune disease was more commonly found in nerve large arterial vasculitis compared to microvasculitis, 68% to 29%. That was especially true for primary vasculitis, including polyarteritis nodosa, eosinophilic granulomatosis with polyangiitis, and microscopic polyangiitis. In contrast, diabetes mellitus was more prevalent in nerve microvasculitis. What was the temporal and clinical characteristics? Nerve large arterial vasculitis occurred with a more acute onset and less chronic onset in large arterial vasculitis compared to microvasculitis. Age of onset in nerve large arterial vasculitis was similar. Nerve microvasculitis had a longer time to diagnosis and a longer time to plateau than nerve large arterial vasculitis. The most common clinical presentation for nerve large arterial vasculitis was an asymmetric polyneuropathy at 48%, whereas for nerve microvasculitis, it was a radiculoplexus neuropathy with proximal limb involvement in 43.8%. The pattern of multiple mononeuropathy, which we think of as classical for vasculitic neuropathy, was infrequently seen in both groups, 18% in large arterial, 14% in microvasculitis. Proximal nerve segment involvement of the lower extremity was seen more frequently in nerve microvasculitis than nerve large arterial vasculitis, 51% to 26%. Pain was very common in both groups. Nerve microvasculitis, 87%, nerve large arterial vasculitis, 87%, nerve microvasculitis, 86%. Autonomic symptoms were much more common in microvasculitis. Systemic involvement. Nerve large arterial vasculitis was associated with systemic symptoms in 61%. It was significantly more than microvasculitis. Systemic symptoms included myalgia, arthralgia, fevers, and occurred more commonly in large arterial vasculitis than microvasculitis. Evidence of other organ involvement by vasculitis was much more common in nerve large arterial vasculitis. Other organ involvement included asthma, sinusitis, pulmonary infiltrates, glomerulonephritis, and rash, and occurred more commonly in nerve large arterial vasculitis compared to microvasculitis. The pathological diagnosis of nerve large arterial vasculitis was associated with systemic vasculitis in 79% compared to 29% in microvasculitis. The pathological diagnosis of microvasculitis was associated with non-systemic vasculitis in 71%. Only 5% of nerve microvasculitis received the clinical diagnosis of primary systemic vasculitis compared to 49% of nerve large arterial vasculitis. Cases of diabetic lumbosacral plexus neuropathy, non-diabetic lumbosacral plexus neuropathy had more microvasculitis than large arterial vasculitis, 31% compared to 2%. So this is another way of looking at this. Nerve large arterial vasculitis was associated with primary systemic vasculitis, where nerve microvasculitis was associated with non-systemic vasculitis. What about the electrophysiology? There was more distal asymmetric polyneuropathy EMG patterns in large arterial vasculitis, 47% versus 18%. There was more radiculoplexus neuropathy EMG patterns in microvasculitis, 44% versus 19%. The nerve microvasculitis group had more proximal involvement based on large motor unit potential and fibrillation potential on needle exam, and obturator nerve distribution, superior gluteal nerve distribution, inferior gluteal nerve distribution. The nerve large arterial vascular group was somewhat more severely affected on nerve conduction abnormalities. The median CMAP was lower, the seral SNAP was lower. Nerve large arterial vasculitis had evidence of systemic involvement by laboratory tests compared to microvasculitis. They had lower hemoglobins, higher white counts, more eosinophilia, higher platelet counts, higher sedimentation rates, higher C-reactive proteins compared to microvasculitis. Nerve microvasculitis had higher CSF proteins in keeping with the pattern of proximal nerve involvement and radiculoplexus neuropathy. Autonomic reflex screen testing showed more impairment, higher CAS scores, and microvasculitis than nerve large arterial vasculitis. The speed of presentation. The speed of presentation is different. Nerve large arterial vasculitis presents more acutely in most cases, where nerve microvasculitis presents more subacutely in most cases. Similarly, the evolution of the disease is longer in microvasculitis than in nerve large arterial vasculitis. As I've mentioned, multiple mononeuropathy is an uncommon clinical pattern in both groups. The pattern of distal asymmetric polyneuropathy is common for nerve large arterial vasculitis, and multiple mononeuropathy really have similar significance, and both are probably the result of ischemic nerve injury. The anatomical pattern is different between nerve large arterial vasculitis and microvasculitis. Nerve large arterial vasculitis presents with distal asymmetric neuropathy in keeping with nerve infarction occurring at watershed zones of vascular supply, mid-arm and mid-thigh levels with summation distally. In contrast, nerve microvasculitis presents as a radiculoplexus neuropathy with more proximal involvement above the mid-arm and the mid-thigh levels. The ischemic injury from microvasculitis does not seem to be restricted to these watershed zones of nerve ischemia. Consequently, there's more proximal findings on examination, electrophysiology, and elevation in CSF proteins in microvasculitis. Severe pain and weakness were common presentations in both types of vasculitis. Autonomic symptoms and findings were more common in the microvasculitis group with higher CAS scores. Widespread autonomic involvement is recognized to be common in both diabetic and non-diabetic lumbosacral radiculoplexus neuropathy, which are forms of microvasculitis. Nonetheless, there are major overlaps in these clinical patterns and features. So there is more evidence of systemic vasculitis and nerve large arterial vasculitis and more evidence of non-systemic vasculitis and nerve microvasculitis. The pattern of radiculoplexus neuropathy was associated with microvasculitis, and as I have mentioned, it's known to occur in diabetic and non-diabetic lumbosacral plexopathies. We found more connective tissue disorders in nerve large arterial vasculitis. Systemic autoimmune disease is more common in nerve large arterial vasculitis, as was primary vasculitis. The mean weight was higher in microvasculitis, and diabetes is more associated with microvasculitis. And this association is likely due to diabetic lumbosacral radiculoplexus neuropathy and microvasculitis. Weight loss occurred in about half patients from both groups. Our study confirms that systemic symptoms are more common in nerve large arterial vasculitis. There are increased rates of non-nerve organ involvement in nerve large arterial vasculitis, including respiratory, renal, skin, joint, and constitutional symptoms compared to microvasculitis. Laboratory markers of systemic illness are more common in nerve large arterial vasculitis, including SED rates, C-reactive proteins, ANAs, white counts, platelet counts, higher eosinophilia. The CSF protein is higher in microvasculitis, and the finding indicates more proximal involvement. So we published this paper in Brain and was put on the cover. So the clinical laboratory electrophysiological manifestations associated diseases of histopathologically confirmed nerve large arterial vasculitis and nerve microvasculitis are distinct with different clinical courses and different outcomes, although there is overlap. Nerve large arterial vasculitis has more acute onset, presents as a distal asymmetric polyneuropathy, is associated with autoimmune disease, with systemic vasculitis, and has abnormal laboratory markers. Nerve microvasculitis presents with more chronic onset, more protracted clinical course, has more proximal nerve involvement with a radiculoplexus neuropathy pattern, is more frequently a non-systemic vasculitis, has more autoimmune involvement, and is associated more commonly with diabetes mellitus. As mentioned this morning by Rick, my father died on July 26th of this year. He did a lot of work on vasculitic neuropathies, and so I just want to put a picture of him up there in the pose of Ramon Cajal. And I want to acknowledge my different co-authors on this, and thank you very much, and I appreciate the invitation to speak here today.
Thank you so much, Jim. Yes, Tony?
Great talk, great paper. How many patients, did you do muscle biopsies at the same time, and did the muscle show vasculitis, either micro or large, and was there a difference?
Yeah, so John Kissel always recommended doing simultaneous nerve and muscle biopsies, and often doing superficial perineal nerve for that reason. So the reality is at Mayo Clinic we rarely do combined muscles, so probably 5% or so we did, but most of them we don't, because we tend to get fairly high diagnostic rates just doing nerve, and so we're usually doing Searle, so we're often not doing muscle with them. Saying all of that, yeah, the ones where we did do muscle often showed the same pathology. Not always, but often. But we didn't specifically look at that in this cohort.
And was there inflammation elsewhere in the nerve biopsies, endoneurium, perineurium? Was it confined to the vessels? And how is microvasculitis different than perivascular inflammation that you might see with other
No, I think that's fundamentally a very important question. So we right now are taking the same cohort. we took, in this cohort, we took people with diagnostic findings of vasculitis and we looked at the clinical features. I'm now going to take the same cohort and pull all those biopsies out and grade them all for all these sort of things you're saying. How common are ischemic features? Are they different? We don't know that. Are the ischemic features different between microvasculitis and large arterial vasculitis? Where is the inflammation? Is it different? So in fact, we are doing that right now. So I can't specifically answer that question, but I will say that perivascular inflammation doesn't get you this diagnosis. As I mentioned early on, we called them diagnostic and large arterial vasculitis much more common than we call them highly suggestive. To be highly suggestive, you have to have vessel wall infiltration and fragmentation. So without that, you don't get it. So perivascular inflammation doesn't get you into this group. No, well, it gets you into the large if you have fibrinoid necrosis with it, but it doesn't get you into the large if you don't have that. You have to have vessel wall destruction and you have to have inflammation. So.
Just one last thing, just kind of interesting, I'm sorry, but with the nerve, peripheral nerve, a brain, what are lacunar infarcts? Are they small infarcts of small vessels? Is that microvasculitis?
Yeah, no, that's an interesting question.
It's not what we often think about in a diabetic with a lacunar infarct.
Oh, no, that's a fascinating question. So in 1970, my father went on, the Mayo Clinic used to pay for a month trip for its consultants to go around Europe to improve themselves so they weren't neurologists in the sticks seeing people, sophisticated people, so they were supposed to go to the Louvre and go to the Colosseum and do these sorts of things to improve their social ability to talk intelligently with Europeans and the people around the world. My father used it as an opportunity to meet experts in neurology around the world. And so he had tea with a Wilhelm Kricke in his garden, and they discussed neuropathology. And the main topic was, are lacunar infarcts in the brain different than infarcts in the nerve? And Kricke's view was, yes, they are quite different, and why are they different? So you're giving me flashbacks, pleasant flashbacks here by bringing that whole subject up.
One last question.
Well, maybe one question wrapped in three questions. First, does size matter when it comes to...
Size always matters, Mazen.
Thank you. And then, can you define that better? But the second question, are all DLSPRN cases of microvasculitis? And third is, where's the window of opportunity for treatment, if any, in microvasculitis?
Oh, you have a lot of questions. Size of what? Size of the vessel, does it matter? Is that what you're asking me? I said size matters. Well, I do think, so many people for years have questioned, does microvasculitis even exist? And I feel strongly that it does, and that it's associated with ischemic injury. I mean, the multifocal fiber loss, perineural thickening, neovascularization, all of these features typical of large arterial vasculitis you absolutely see in the microvasculitis. I mean, I think this absolutely exists. I think the size does seem to matter in that the clinical patterns are different. I mean, why do you get this radicular plexus neuropathy pattern with a microvasculitis and you don't get that pattern typically with a large arterial vasculitis? I mean, that has to mean something. So, treatment implications, how quickly do you treat? I think that's a very important question. So we did a controlled trial with IV methylprednisolone and diabetic lumbosacral plexopathy on microvasculitis, and we insisted they had to be within the first 6 months, and it didn't, to the primary endpoint, didn't show a difference because I think we got them in too late. It's hard to identify these people early enough, but it's a monophasic illness. Honestly, large arterial vasculitis is probably also a monophasic illness. So, nobody's ever done a controlled trial on that disease. And sorry, what was your third question?
Well, I think, are there cases of DLSPRN where you think that it's not microvasculitis, it's metabolic, or you think this is all microvasculitic?
I never say always, so sure. I think, yes, there are in this cohort some large arterial vasculitis that presents as with more proximal involvement and more of a radicular plexus neuropathy pattern. So life is complicated.
Thank you, Jim. Thank you to the speakers.
Thank you.
And